Every living thing you've ever known has died. Every person, every animal, every plant. From the smallest bacteria to the largest whale, life follows one unbreakable rule. You are born, you age, and eventually you stop. Except for one. Somewhere in the world's oceans, drifting in darkness, there's a creature no bigger than your pinky nail that discovered something impossible. When it reaches the end of its life, when its body begins to fail and death approaches, it doesn't die. It reverses. Its cells don't just stop aging, they go backward. Muscle becomes stem cell. Specialized tissue becomes generic. The adult creature dissolves into a juvenile form and starts over. And it can do this again and again, as many times as it needs to. We call it Turritopsis dohrnii, the immortal jellyfish. But here's what keeps scientists awake at night. This isn't some genetic anomaly, some freak accident of evolution. This is a deliberate biological program. The jellyfish has a switch. When threatened with death, it flips that switch and runs time backward at the cellular level, which means immortality isn't impossible. It's been solved. For 500 million years, this creature has been doing what we can only dream of, choosing not to die. Tonight we're going to trace how this works, from the molecular signals that trigger reversal, to the cells that remember how to become young again, to what this means for aging, death, and whether immortality might be hiding in our own DNA. Because if one animal found the secret, the question isn't whether immortality is possible, it's whether we can learn the language. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a jellyfish in the open ocean. It's night. The water is black, cold, infinite in every direction. The jellyfish drifts, a small translucent bell, maybe 4 millimeters across, trailing 90 delicate tentacles into the void. It glows faintly, bioluminescence pulsing through its body in waves. Blue-green light, then darkness, then light again. A heartbeat made visible. This is Turritopsis dohrnii, and right now, it's hunting. Tiny crustaceans, zooplankton smaller than grains of sand drift past. When one brushes a tentacle, specialized cells called cnidocytes fire, microscopic harpoons loaded with venom. The prey is paralyzed instantly. The tentacle contracts, drawing the prey toward the mouth at the center of the bell. The jellyfish feeds. It's been doing this for weeks, maybe months, growing slowly, maturing. Its gonads have developed, visible through the translucent bell as bright orange structures along the radial canals. It's an adult now, ready to reproduce, ready to release eggs or sperm into the water and fulfill the biological imperative that drives all life, create the next generation, then die. But something is wrong. Maybe it's injury. A predator took a bite, tore through the delicate bell tissue. Maybe it's starvation. Food has become scarce, and the jellyfish is metabolically exhausted. Maybe it's temperature stress or infection, or simply the accumulated damage of living. Whatever the cause, the jellyfish is dying. Its movements slow, the bioluminescence flickers irregularly, the tentacles begin to retract. In any other animal, this would be the beginning of the end. Cells would shut down, tissues would fail, death would follow. But this jellyfish does something no other animal on Earth can do. It sinks, down through the water column, away from the light, toward the ocean floor. When it reaches the bottom, sand, rock, coral, whatever solid surface exists below, it attaches itself. The bell collapses. The tentacles are reabsorbed. The adult body, the sexually mature medusa that was hunting and reproducing just hours ago, begins to dissolve. From the outside, it looks like death. Tissue breaking down, structure disintegrating, the creature melting into formless mass. Except the cells aren't dying, they're transforming. Inside that collapsing mass, something extraordinary is happening at the molecular level. Muscle cells, specialized cells that contract to propel the jellyfish through water, are de-differentiating, losing their identity, forgetting what they were. Nerve cells, The neurons that coordinate movement and feeding are doing the same. Cell by cell, the entire organism is erasing its own adulthood. And then, from that dedifferentiated tissue, new structures begin to form. Not the structures of an adult jellyfish. The structures of a polyp, the juvenile stage that jellyfish grow from when they first develop. Small tentacles emerge. A stalk forms. A mouth opens. Within days, what was an adult floating in? Open water has become a juvenile attached to the seafloor. The jellyfish has gone backward in time. This process is called transdifferentiation, and until 1996, most biologists didn't think it was possible in complex animals. Cells differentiate during development, becoming muscle, nerve, skin, whatever the body needs, and that's supposed to be permanent. A one-way door. You can't unring the bell. Except Turritopsis dohrnii does exactly that. It rings the bell backward, and here's the truly impossible part. It can repeat this process multiple times, potentially forever. The polyp will live on the seafloor for weeks or months, feeding and growing. Then, when conditions are right, it will bud. Small medusae will form on its body and detach, swimming away as new jellyfish. One of those medusae will mature, reproduce, age, and when death approaches again, it will sink, transform, and start over. Birth, growth, maturity, death, rebirth, an infinite loop. We've known about this jellyfish since the late 1800s, but we didn't understand what it was doing until Japanese researchers observed the transformation in a lab in the 1990s. Since then, scientists have watched it happen dozens of times. Same individual jellyfish, dying and reversing over and over in controlled conditions. In theory, barring predation or disease, Turritopsis dohrnii could live indefinitely. It is, as far as we know, biologically immortal. But immortality raises questions that science fiction has asked for centuries. What does it mean to live forever? Is the polyp the same individual as the medusa, or is this a kind of death and resurrection? Do the cells remember? And most urgently for us, could we do this? Because we're not as different from jellyfish as you might think. We're both animals. We both develop from a single fertilized egg that differentiates into specialized cell types. We both age. We both die. The molecular machinery of life, DNA, proteins, cellular signaling, is fundamentally the same. If a jellyfish can flip a switch and reverse aging at the cellular level, then that switch exists in biology, which means it might exist in us. Tonight we're going to follow the science of immortality from the ocean floor to the genetic code. We'll trace how a jellyfish's cells remember how to become young, why most animals can't do this, and what happens when researchers try to trigger the same process in human cells. We'll explore the paradox of infinite life, the evolutionary trade-offs that make immortality rare, and the strange fact that your body already contains cells that refuse to die, cancer. Because understanding how Turritopsis dohrnii cheats death might not just explain immortality, it might explain mortality, why we age at all, why our cells have built-in expiration dates, and whether dying is a feature of life or a bug we could someday fix. Let's begin where all impossible things begin, with a creature small enough to fit on your fingertip glowing in, the dark, refusing to obey the one rule we thought was unbreakable. The discovery of biological immortality happened by accident, in a laboratory where nobody was looking for it, University of Lecce, Italy. A marine biology student named Christian Sommer was studying hydrozoans, small jellyfish-like creatures, for his undergraduate thesis. He'd collected several specimens of Turritopsis dohrnii from the Mediterranean and was keeping them in petri dishes, observing their life cycles, taking notes on feeding behaviour, reproduction, the standard cataloguing work of marine biology. The jellyfish were dying, which was expected. Captivity is stressful. The animals were small, delicate, difficult to maintain. Sommer watched them decline, bell tissue deteriorating, movement slowing, the familiar trajectory toward death. He made notes. He prepared to collect new specimens. But then something happened that shouldn't have been possible. The dying jellyfish didn't die. Instead, they sank to the bottom of their containers, attached to the glass or plastic surface, and their bodies began to collapse. The bell structure dissolved, tentacles retracted and were reabsorbed. Within 24 to 36 hours, what had been a free-swimming medusa, an adult jellyfish, had transformed into a blob of undifferentiated tissue stuck to the bottom of the dish. Sommer assumed this was an unusual form of death, some kind of decay process he hadn't seen before. But he kept watching, because that's what scientists do. They document everything, even the unremarkable. And then, from that blob of tissue, something began to grow. Not the decomposition you'd expect from a dead organism. New structures, tiny tentacles, maybe 8 or 10 of them, emerging from the mass. A stalk forming, a mouth opening at the centre. Within a few more days, what he was looking at wasn't a dead jellyfish at all. It was a polyp. The juvenile form that jellyfish grow from during their early development. The stage that comes before the medusa, not after. The jellyfish had gone backward. Sommer brought this to his advisor, Fernando Buero, a professor of marine biology. Together, they repeated the observation. They stressed the jellyfish deliberately, starving them, injuring them, creating conditions that would normally lead to death. And every time, the same thing happened. The medusa would sink, collapse, and transform into a polyp. Not some of them, all of them. It was reproducible. It was real. According to research published by Buero and his colleagues in 1996, they documented the complete transformation process in laboratory conditions. They described it as ontogeny reversal, a return to an earlier developmental stage. But what they'd actually discovered was something far stranger. An animal that could escape death by becoming young again. The findings were published. The scientific community took notice, but cautiously. Immortality is the kind of claim that sounds like pseudoscience or mythology, not rigorous biology. There had to be an explanation that didn't involve breaking the fundamental rules of aging and death. But the jellyfish didn't care about skepticism. It kept doing the impossible over and over in labs around the world. By the early 2000s, researchers in Japan, Spain, and the United States had confirmed it. Turritopsis dohrnii could reverse its life cycle, not just once, but multiple times. Shin Kubota, a marine biologist at Kyoto University, became particularly obsessed with the creature. He kept colonies of them for years, inducing the transformation repeatedly in the same individuals. He documented dozens of reversals in single specimens. In one case, according to Kubota's observations published in 2011, he watched the same jellyfish cycle through medusa and polyp stages more than 10 times over the course of 2 years. Each time it reached maturity and was stressed, temperature change, food deprivation, physical damage, it would reverse. And each time, it would regenerate perfectly, emerging as a healthy polyp that would eventually bud new medusae and start the cycle again. 10 times. The same individual, dying and reversing over and over. If you remove predation, if you protect this jellyfish from being eaten, and you remove disease and you give it enough food and stable conditions, there's no obvious endpoint, no built-in expiration. The jellyfish just continues, looping through its life cycle indefinitely. This is what biologists mean when they say biological immortality. Not that the organism can't die. It absolutely can, from injury, predation, disease, environmental catastrophe. But it doesn't have a programmed death. There's no aging clock counting down, no cellular mechanism that says your time is up. For Turritopsis dohrnii, death is optional, a choice the organism makes only when survival as an adult is no longer viable. But here's what made scientists deeply uncomfortable about this discovery. Aging is supposed to be universal. It's one of the few things that every complex organism shares. Bacteria can theoretically divide forever if conditions are perfect, but animals age. Cells accumulate damage, DNA degrades, proteins misfold, tissues wear out, eventually the system fails. That's biology. That's thermodynamics. That's the second law, entropy in action. Everything moves toward disorder. Except this jellyfish had found a way to rewind. The question that followed wasn't just how does it do this? It was deeper, more unsettling. If one animal solved mortality, why didn't every animal evolve the same solution? Because let's be clear, evolution doesn't care about fairness or morality. It cares about reproductive success. If a trait increases your chances of passing on genes, Selection favours it. Immortality seems like the ultimate advantage. Infinite time to reproduce, infinite chances to adapt. Why isn't every creature on Earth doing this? The answer, as it turns out, is that immortality has a cost, and understanding that cost requires understanding what aging actually is and why it exists in the first place. But before we get there, we need to go deeper into the mechanism itself. Because what Teratopsis is doing, this cellular time reversal, isn't magic. It's a biological program, a specific sequence of molecular events that can be studied, mapped, and potentially replicated. The jellyfish isn't defying the laws of nature. It's using them in a way we didn't know was possible. So let's follow the process from the outside in, from the moment a dying jellyfish makes the decision to reverse, down through the tissues and cells, into the nucleus where genes are switching on and off in precise patterns, rewriting the identity of every cell in the organism. Because if we can understand the language the jellyfish uses to tell its cells go backward, we might be able to speak that language ourselves. The transformation begins with stress. Not just any stress, mortal stress. The kind of damage or deprivation that signals to the organism. Survival as an adult is failing. In laboratory conditions, researchers can trigger the reversal artificially by starving the jellyfish, changing water temperature dramatically, or physically damaging the bell tissue. In the wild, it's probably similar triggers: injury from predators, environmental shock, starvation. But here's what's crucial. The jellyfish has to sense that stress at the cellular level and make a coordinated decision to reverse. This isn't a passive collapse. It's an active program, requiring energy and organization. According to research by Maria Pia Milieta at Texas A&M University and her colleagues published in 2007, the process follows a specific sequence. First, the medusa stops feeding. Its movement becomes erratic. Then it sinks and attaches to a substrate. This part seems critical. The jellyfish needs a stable surface to anchor itself during transformation, similar to how the polyp stage is always sessile, attached to rock or coral or seafloor. Once attached, the bell begins to collapse inward. The outer layer of cells, the epidermis, starts to thicken. The inner layer, the gastrodermis lining the gut cavity, does the same. The mesoglea, the jelly-like substance between the two layers, begins to break down and reorganize. Tentacles retract. In adult Teratopsis, there can be 80 to 90 tentacles trailing from the bell margin. During reversal, these are systematically reabsorbed, not shed or lost, but pulled back into the body and broken down. The cells that made up those tentacles don't die. They're recycled. The gonads, the reproductive organs that were producing eggs or sperm, stop functioning and regress. Specialized reproductive cells dedifferentiate back into unspecialized cells. The whole organism is erasing its sexual maturity. Within 24 to 48 hours, what was a free-swimming, sexually reproducing adult has become a sessile, a sexually reproducing polyp. Different body plan, different cell types, different life strategy. This is ontogeny reversal, literally going backward through developmental stages. And it shouldn't work because development is supposed to be a one-way process. When you're an embryo, your cells differentiate. A stem cell becomes a muscle cell, or a nerve cell, or a skin cell. That specialization is locked in by changes to the cell's DNA expression, not the DNA sequence itself, but which genes are turned on or off, which parts of the genome are accessible. This is called epigenetics, and it's how your body knows that a liver cell should act like a liver cell and not suddenly start being a brain cell. The genetic instructions are the same in every cell. You have the same DNA in your liver as in your brain, but the expression is different, locked into specific patterns. Cell differentiation is like a ball rolling downhill into a valley. Once it's there, it's stable. It takes energy to push it back uphill and over into a different valley. In most animals, that energy isn't available. Once a cell differentiates, it stays differentiated for life. But Teratopsis has found a way to push the ball back uphill. To unlock the differentiated state, erase the cell's specialized identity, and allow it to become something else. This process is called transdifferentiation, when one specialized cell type becomes another specialized type directly, like a muscle cell becoming a nerve cell. But what Teratopsis does is even more dramatic. It's dedifferentiation, specialized cells reverting to a stem-like state, losing their identity entirely, and then redifferentiating into the cell types needed for a polyp. and this happens across the entire organism simultaneously. Every muscle cell in the bell, every nerve cell in the nerve net, every kiernostocyte, the specialized stinging cell unique to jellyfish. All of them, at the same time, following a coordinated program to become young again. The scale of this coordination is staggering. Your body contains roughly 37 trillion cells, all differentiated into about 200 different cell types. Imagine all of them simultaneously forgetting what they are, reverting to stem cells, and then reorganizing into a younger version of you. Not just some cells, all of them. Every tissue, every organ working in concert to rebuild the body from scratch while you're still alive inside it. That's what Turritopsis does. And the fact that it works, that the organism doesn't just dissolve into chaos but instead executes a precise developmental program in reverse, means there are genetic instructions for this process. Genes that say when to de-differentiate, genes that coordinate the transformation, genes that know how to rebuild a polyp from the raw material of a medusa. We're only beginning to understand what those genes are. The jellyfish genome was partially sequenced in recent years, and researchers are now comparing gene expression in normal medusae versus medusae undergoing transdifferentiation. Early results, according to studies from 2022 by researchers at the University of Oviedo in Spain, show massive upregulation of genes related to pluripotency, the ability of a cell to become multiple different cell types. These are the same genes that are active in embryonic stem cells, the genes that allow a fertilized egg to become an entire organism. Turritopsis appears to be reactivating its embryonic genetic program in adulthood, essentially telling its cells, go back to the beginning, remember what you were before you specialized, we're starting over. But activation of pluripotency genes alone isn't enough. You also need to suppress the genes that maintain differentiated identity, coordinate the timing across millions of cells, prevent the whole thing from collapsing into cancer, because uncontrolled differentiation is basically what cancer is, and then re-establish a new body plan. The genetic choreography required for this is extraordinarily complex, and yet somewhere in the jellyfish's DNA, in the regulatory networks that control gene expression, there's a switch. When thrown, it initiates the entire cascade. The question that keeps researchers awake at night is simple. Do we have that switch? To understand if we have that switch, we need to understand what the switch actually is, not metaphorically, but mechanically. What genes are involved? What proteins do they produce? What signals tell a cell that it's time to forget everything it knows about being a muscle cell or a nerve cell and become something unspecialized again? The answer starts with a group of proteins that most people have never heard of, but that every cell in your body relies on to maintain its identity. They're called transcription factors, and they're essentially the managers of your genome. Your DNA contains about 20,000 genes, but not all of them are active in every cell. A liver cell uses liver genes, a brain cell uses brain genes. Transcription factors are proteins that bind to specific regions of DNA and either turn genes on or make them accessible for activation. They're what makes a stem cell different from a skin cell, even though both contain the exact same genetic information. In the 2000s, a Japanese researcher named Shinya Yamanaka made a discovery that would win him the Nobel Prize and fundamentally change how we think about cell identity. He found that you could take an adult cell, a fully differentiated skin cell from a mouse, and turn it back into a stem cell by introducing just 4 transcription factors. 4 proteins. That's all it took to erase years of specialization and return the cell to an embryonic-like state. These became known as the Yamanaka factors: OCT4, SOX2, KLF4, and c-MYC. 4 proteins that, when expressed together in an adult cell, reprogram its identity. The cell forgets it was skin. It becomes pluripotent again, capable of becoming any cell type in the body. This was revolutionary because it proved that differentiation isn't permanent. The lock can be picked. The valley that the ball rolled into can be escaped if you have the right molecular tools. And here's where it gets interesting. When researchers analysed gene expression in Turritopsis dorni during transdifferentiation, they found that the jellyfish is doing something remarkably similar. According to studies published in 2022 by Maria Pascual Torner and colleagues at the University of Oviedo, the immortal jellyfish shows significant upregulation of genes related to pluripotency during its transformation. Not the exact same genes as the Yamanaka factors, jellyfish and mammals diverged evolutionarily hundreds of millions of years ago, so the specific proteins are different, but functionally equivalent genes. Genes that destabilize cell identity and promote a return to a stem-like state. The jellyfish has its own reprogramming cocktail, its own set of molecular keys that unlock differentiation, but it's doing something Yamanaka's technique doesn't do. It's coordinating this reprogramming across an entire organism, not just isolated cells in a lab dish. Every cell in the jellyfish's body has to de-differentiate at roughly the same time, and then redifferentiate in a coordinated way to build a polyp instead of a medusa. That requires communication between cells, signaling pathways that say when to start, when to stop, what to become. One of the key signaling systems involved appears to be the WNTS pathway, a communication network that's ancient in evolutionary terms. It exists in everything from jellyfish to humans, and it plays a crucial role in development, telling cells where they are in the body, what structures to form, how to organize into tissues and organs. During turritopsis transdifferentiation, WNT signaling genes are activated. This makes sense. If the organism is essentially rerunning a developmental program, it needs the same molecular machinery that guides development in the first place. The jellyfish is telling its cells, we're rebuilding, follow the developmental map, but go to the polyp section, not the medusa section. There's also evidence of autophagy ramping up dramatically. Autophagy is cellular self-eating, the process by which a cell breaks down its own components, recycling proteins and organelles that are damaged or no longer needed. Is how your cells clean house. During the transformation, turritopsis cells are essentially digesting themselves in a controlled way, breaking down the specialized structures of adult cells and freeing up the molecular building blocks to construct new, different cells. Imagine renovating a house while you're still living in it, but instead of just redecorating, you're tearing down walls, melting the bricks back into clay, and rebuilding the entire structure into a different floor plan. That's what every cell in the jellyfish is doing simultaneously. And somehow, the organism doesn't die in the process. The metabolism continues, the cells stay alive, the genetic program is precise, enough that chaos doesn't take over. This precision is what separates turritopsis from cancer. Because here's the uncomfortable truth. Cancer cells are also immortal. They've also figured out how to escape the normal limits on cell division and lifespan. But they do it in a way that destroys the organism. They grow uncontrollably, ignore signals from neighbouring cells, consume resources without contributing to the body's function. Cancer is cellular immortality without coordination. Turritopsis is cellular immortality with coordination. The difference is regulation. In normal aging, your cells have built-in limits on how many times they can divide. This is controlled partly by structures called telomeres, repeating DNA sequences at the ends of chromosomes that get shorter each time a cell divides. Think of them like the plastic tips on shoelaces. They protect the chromosome ends from damage, but they wear down with use. After a certain number of divisions, the telomeres become critically short, and the cell either stops dividing or dies. This is called the Hayflick limit, and it's one of the fundamental mechanisms of aging. Cancer cells bypass this limit by reactivating an enzyme called telomerase, which rebuilds telomeres. The cell can now divide indefinitely. It's become immortal. But it's also become selfish, no longer cooperating with the rest of the body, just reproducing without limit. According to research on turritopsis published in 2020, the jellyfish also shows high telomerase activity during and after transdifferentiation. Its cells are rebuilding their telomeres, resetting the division clock. But unlike cancer, this is happening in a coordinated, regulated way across the entire organism. The cells aren't going rogue. They're following a program. It's the difference between a controlled demolition and a building collapse. Both involve structures coming down, but one is intentional and organised, the other is catastrophic. So turritopsis has solved a problem that medicine has been wrestling with for decades. How to make cells young again without making them cancerous. How to reactivate the machinery of youth, pluripotency factors, telomerase, developmental signalling, while maintaining the regulatory networks that keep cells cooperating as part of a functional organism. This is why the immortal jellyfish is so important. It's not just a curiosity, a weird animal that does a weird thing. It's a proof of concept. It demonstrates that biological immortality and multicellular organization are compatible. You can have both if the regulation is sophisticated enough. But that raises the next question. Could we do this? Could human cells be reprogrammed the way jellyfish cells are? Could we trigger dedifferentiation and redifferentiation in our own tissues, resetting the aging clock without causing cancer? The short answer is, we're trying, and we've already succeeded in limited ways. Yamanaka's induced pluripotent stem cells, the adult. Cells reprogrammed back to stem cells using those 4 transcription factors are already being used in research and medicine. Scientists can take a skin cell from a patient, turn it into a stem cell, and then coax it to become a heart cell, or a neuron, or insulin-producing pancreatic cells. This is regenerative medicine, growing replacement tissues from a patient's own cells, but that's in a lab dish. Isolated cells, carefully controlled conditions. The question is whether you can do it in a living organism, in a whole body the way Turritopsis does. In 2016, researchers at the Salk Institute tried. They took mice that were engineered to age rapidly, a genetic condition that mimics accelerated aging, and they introduced the Yamanaka factors directly into the animal's tissues. Not constantly, but in pulses. Brief periods of reprogramming, then stopping, then repeating. The mice got younger. Not all the way back to youth, but measurably. Their tissues showed reduced signs of aging. Their cells functioned better. Some of the damage from aging was reversed, but there was a problem. If the reprogramming was too strong or lasted too long, the mice developed tumours. The cells lost their identity too completely and started behaving like cancer. The experiment walked a razor's edge between rejuvenation and catastrophe. According to a study published in Cell in 2016 by Alejandro Ocampo and colleagues, the key was finding the right dose and timing. Partial reprogramming, just enough to reset some aging markers without erasing cell identity entirely, could extend lifespan and improve health without causing cancer. But the window was narrow. Since then, other labs have refined the technique. They've tried different combinations of factors, different delivery methods, different timing protocols. The results are promising, but still experimental. We're learning to speak the language of cellular reprogramming, but we're not fluent yet. And here's where Turritopsis becomes the teacher. Because the jellyfish isn't doing partial reprogramming, it's doing complete dedifferentiation and redifferentiation across its entire body, repeatedly, without developing cancer. Whatever regulatory mechanisms it's using, whatever genes are keeping the process organized and safe, those are the mechanisms we need to understand. Recent genomic studies are starting to identify candidates, genes involved in cell cycle control, genes that suppress tumor formation, genes that coordinate developmental timing. The jellyfish genome is being mapped, compared to other cnidarians that can't do this trick, compared to our own genome to see what's shared and what's unique. One hypothesis is that Turritopsis has enhanced DNA repair mechanisms. Aging and cancer both involve accumulated DNA damage, mutations that pile up over time. If the jellyfish can repair that damage more effectively during its transformation, it would explain how the process remains stable. The cells are being wiped clean of errors, reset to a pristine state. Another hypothesis focuses on the jellyfish's relatively simple body plan. It doesn't have a brain, it doesn't have organs in the way we think of them. Its nervous system is a diffuse net, not a centralized processor. Maybe that simplicity makes total body reprogramming easier. There are fewer cell types to coordinate, fewer specialized structures that have to be preserved. If that's true, it suggests that immortality might be harder for complex animals like us. Our brains alone contain 86 billion neurons, each one a specialized cell with unique connections to thousands of other neurons. Those connections are what make you you, your memories, your personality, your sense of self. If you de-differentiated all your neurons back to stem cells, would you still be the same person when they redifferentiated? Or would you wake up as someone else with a blank mind? This is the ship of Theseus problem, but biological. If you replace every cell in your body, are you still you? Turritopsis doesn't seem to worry about this. The polyp that emerges from a transformed medusa doesn't have the medusa's experiences or behaviours, at least not that we can measure. It's effectively a new individual, even if it's genetically identical to the one that died. The jellyfish achieves immortality by becoming something else, by letting go of what it was. For humans, that might not be acceptable. We want to live longer, but we want to remain ourselves. We want continuity of consciousness, preservation of memory, identity that persists. Turritopsis trades identity for survival. It's not clear if we'd be willing to make that trade, but maybe we won't have to. Maybe there's a middle path. Partial reprogramming that resets aging without erasing memory. Rejuvenation without loss of self. That's the hope driving research in this field. And Turritopsis is the roadmap. An existence proof that biology can solve mortality if the mechanisms are understood and controlled. But to understand why those mechanisms are so rare, why Turritopsis is one of the only animals that does this, we need to step back and ask a bigger question. Why does aging exist at all? If immortality is biologically possible, why did evolution build death into almost every living thing? The answer has to do with trade-offs, competition, and the brutal economics of reproduction in a finite world. Because it turns out, living forever has a cost. And for most species, that cost is too high. Here's the paradox. Evolution should favour immortality. Natural selection rewards anything that helps an organism survive and reproduce. More survival time means more opportunities to pass on genes. If a mutation appeared that allowed an animal to live indefinitely, that animal would have infinite reproductive potential. Its genes would spread. Immortality should be everywhere, but it's not. Aging and death are nearly universal among complex animals. We're born, we grow, we reproduce, we decline, we die. The pattern repeats across species, across phyla, across kingdoms of life. Even organisms that can theoretically live for centuries, certain trees, some species of clams, still show signs of senescence eventually. Cells break down, systems fail, nothing lasts forever, except Turritopsis, and a handful of other organisms that have found loopholes in the aging program. So what happened? Why did evolution build death into almost everything, when immortality seems like the ultimate advantage? The answer isn't simple, and it took biologists decades to work out, but it comes down to this. Death isn't a bug in the system. It's a feature, and in most ecological contexts, it's a feature that makes species more successful, not less. Let's start with the most basic constraint. Resources are finite. Imagine a population of immortal animals on an island. They reproduce. Their offspring reproduce. Population grows. Eventually the island runs out of food. Competition becomes intense. The old individuals, the ones who have been alive for decades or centuries, are occupying space and consuming resources, but they're no longer reproducing as efficiently. Their bodies might be worn down even if they're not aging in the traditional sense. They're still competing with their own descendants for the same food, the same territory. In this scenario, a mutation that causes death after reproduction might actually be beneficial for the species. The old generation dies off, freeing up resources for the young. The population as a whole reproduces faster, adapts more quickly, because generations are turning over. New genetic combinations are tested. Evolution can respond to changing environments. This is part of what's called the disposable soma theory, proposed by biologist Thomas Kirkwood in the 1970s. The idea is that organisms have finite energy and resources. They can invest in maintenance and repair, keeping the body young indefinitely, or they can invest in reproduction, but they can't do both perfectly. There's a trade-off. For most organisms, evolution has favoured reproduction over immortality. Pour your energy into making offspring, not into living forever. Once you've reproduced, your evolutionary job is done. Staying alive after that point offers diminishing returns. You're competing with your own children and grandchildren. Better to age and die, making room for the next generation. According to Kirkwood's theory, this is why we age. Not because aging is inevitable from a physics standpoint, but because evolution never bothered to build perfect repair systems. The genetic programs that would keep us young forever exist in principle. Turritopsis proves that, but for most species those programs are too expensive. The energy cost of maintaining immortality is higher than the reproductive benefit it provides. But here's the catch. This logic only works in certain ecological contexts. It works if you're living in a stable environment with consistent predation, where most individuals die young from external causes anyway. If a mouse's average lifespan in the wild is 6 months because of owls and foxes, there's no evolutionary pressure to build a body that could last 10 years. The mouse will be eaten long before aging becomes relevant. So evolution invests in fast reproduction, not long-term maintenance. But what if you're in an environment with low predation? What if you're small enough, or well defended enough, or living in a niche where death by external causes is rare? Then suddenly immortality starts to make sense again. If you can avoid being eaten, if you can survive indefinitely, then the costs of maintaining your body are worth it. You can reproduce continuously, year after year, accumulating offspring over an extended lifetime. This might be part of why Turritopsis evolved its ability. Jellyfish are soft-bodied, vulnerable, and often eaten. But they're also incredibly abundant. They reproduce prolifically, and in certain environments, protected bays, areas with low predator density, a jellyfish might reasonably survive for extended periods if it can avoid being consumed. In that context, having a reset button makes evolutionary sense. If you're injured or starving, or facing environmental stress that would kill you as an adult medusa, transform into a polyp. Ride out the bad conditions as a sessile, low-energy form. Then when things improve, bud off new medusae and try again. It's not true immortality in the sense of living indefinitely as the same individual. It's more like having a backup save file. When the game gets too hard, reload from an earlier checkpoint and try a different strategy. But this only works because jellyfish have simple bodies. They can afford to completely reorganize themselves. Mammals can't. We have brains that take years to develop and decades to accumulate experience. We have immune systems that learn and remember pathogens. We have complex organ systems, hearts, livers, kidneys, that can't just be de-differentiated and rebuilt without killing us in the process. Our complexity is both our strength and our prison. This is the other reason immortality is rare. It's mechanically hard to achieve in a complex body. Think about what would be required to reset a human body the way Turritopsis resets itself. You'd need to de-differentiate trillions of cells simultaneously, coordinate their transformation, rebuild every organ system, and somehow preserve the brain's information content, your memories, your learned skills, your sense of self, while the neurons themselves are being replaced. The technical challenges are staggering, and evolution rarely invests in building systems that complex unless the payoff is enormous. For most animals, the payoff isn't there. It's cheaper, evolutionarily speaking, to reproduce and die than to build the cellular machinery for total body regeneration. But some organisms did find middle paths, ways to live longer without full immortality. Certain species of rockfish can live over 200 years. Bowhead whales can reach similar ages. Greenland sharks might live 400 years or more. They're not immortal. They do age slowly, but they've drastically extended their lifespans compared to similar-sized relatives. How? By investing in better DNA repair, more robust cellular maintenance, slower metabolisms. They pay the cost in other ways. Many long-lived species reproduce slowly, mature late, have fewer offspring per generation. They're playing a different evolutionary strategy: quality over quantity, endurance over speed. And then there are organisms that went even further. The naked mole rat, a bizarre rodent that lives in underground colonies in East Africa, shows almost no signs of aging for most of its life. It can live over 30 years, 10 times longer than similar-sized mice, and it doesn't follow the normal pattern of increasing mortality with age. A 10-year-old naked mole rat is just as likely to survive the next year as a 2-year-old. According to research published in 2018 in eLife by Ruby, Smith, and Buffenstein, naked mole rats appear to have escaped Gompertz's law, the mathematical rule that describes how mortality risk increases exponentially with age in most animals. For humans, your risk of death roughly doubles every 8 years after age 30. For naked mole rats, risk stays flat. They don't get more likely to die as they get older. They achieve this through a combination of mechanisms: extremely efficient DNA repair, high expression of proteins that prevent misfolded proteins from accumulating, strong cancer resistance. Naked mole rats almost never develop tumours. They've invested heavily in maintenance, but they also live in an unusual ecological niche: underground, in stable temperatures, in social colonies where most individuals don't reproduce. The evolutionary pressures are different. For a non-breeding worker, There's no trade-off between reproduction and longevity. All of its evolutionary value comes from helping the colony survive long-term. So natural selection favoured mechanisms that keep workers alive and functional for decades. Different ecology, different solution. But still not true immortality. Naked mole rats do eventually age and die. They've extended the warranty, not eliminated expiration. Teratopsis went further. It found a way to reset entirely, but it paid a price for that ability. The price is simplicity. Jellyfish are among the simplest animals with true tissues. They have no brain, no centralized organs, no complex developmental programs that can't be easily reversed. Their flexibility comes from their lack of specialization. We can't go back to that level of simplicity. Complexity is built into us. Our brains alone took 500 million years of evolution to develop, and they're what make us capable of asking these questions in the first place. We're not going to trade consciousness for immortality, but maybe we don't have to. Maybe there's a way to have both. Because here's what the naked mole rats and the rockfish and the Greenland sharks are teaching us. Aging isn't monolithic. It's not one single process with one single cause. It's a collection of processes: DNA damage accumulation, protein misfolding, cellular senescence, telomere shortening, mitochondrial dysfunction, stem cell exhaustion, and different organisms have solved different parts of the puzzle. Naked mole rats solved protein quality control. Rockfish solved DNA repair. Teratopsis solved cellular dedifferentiation. No single organism has solved everything, but the solutions exist in nature, distributed across different species. What if we could collect those solutions, take the best parts of each long-lived organism's toolkit and combine them? This is where the field of longevity research is heading. Not trying to copy Teratopsis exactly, we're not going to turn into polyps, but identifying the specific mechanisms that different species use to extend lifespan or escape aging and figuring out how to apply those mechanisms to humans. Some of this is already happening. Drugs that activate the same metabolic pathways that extend lifespan in worms and flies are in clinical trials for humans. Gene therapies that boost DNA repair or clear senescent cells are being tested. Compounds that mimic the effects of caloric restriction, one of the most reliable ways to extend lifespan across species, are in development. None of this will make us immortal, at least not in the foreseeable future, but it might make us live longer, healthier, with less of the decline that characterizes old age. And if we understand Teratopsis deeply enough, if we map every gene involved in its transformation, every signaling pathway, every regulatory mechanism, we might learn how to trigger partial reprogramming in specific tissues, reset your heart cells without affecting your brain, rejuvenate your immune system without disrupting your gut, targeted cellular renewal, not total body transformation. That's the dream. Immortality isn't the goal for most researchers. The goal is extending health span, the number of years you're healthy and functional, not just lifespan. Because living to 150 doesn't mean much if you spend the last 50 years frail, sick, and in pain. But to get there, we have to understand not just how Teratopsis does what it does, but why most animals don't. Why evolution built aging into us. What trade-offs were made, and whether those trade-offs still apply in the modern world. Because here's the thing, humans have changed the evolutionary equation. We don't die from predation anymore. We're not competing for scarce resources in the same way our ancestors were. We've built medicine, agriculture, technology that insulates us from many of the pressures that shaped our biology. In the environment we evolved in, small tribes, high predation, limited food, it made sense to age and die after reproducing. But in the environment we live in now, those old evolutionary rules might not apply. We might be stuck with aging not because it's still adaptive, but because evolution hasn't had time to catch up. If that's true, then maybe we can do what evolution hasn't: redesign the system, take conscious control of our own biology, and build the repair mechanisms that evolution decided weren't worth the investment. It's a radical idea, and it comes with enormous ethical questions. Should we extend human lifespan? What would that do to society, to population growth, to resource consumption, to inequality? If immortality becomes possible, who gets access? Do we want to live in a world where people can choose not to die? These aren't hypothetical questions anymore. They're becoming real as the science advances, and Teratopsis drifting in the ocean, indifferent to philosophy and ethics, is showing us that the biology is possible. The question is whether we have the wisdom to use it. There's an uncomfortable truth at the heart of immortality research, one that scientists don't like to talk about in public, but whisper about in labs late at night. Cancer has already solved immortality, not in the way Turritopsis has with coordination and control, but cancer cells are truly immortal. They divide indefinitely. They ignore the signals that tell normal cells to stop. They've bypassed every checkpoint, every failsafe, every mechanism that limits cellular lifespan, and they're extraordinarily good at it. The most famous immortal cells in biology aren't from a jellyfish. They're from a woman named Henrietta Lacks who died of cervical cancer in 1951. Before she died, doctors at Johns Hopkins Hospital took a sample of her tumour without her knowledge or consent. Those cells were cultured in a lab, and something remarkable happened. They didn't die. Normal human cells in culture divide a few dozen times and then stop. They hit the Hayflick limit, their telomeres run out, and they enter senescence, a permanent state of growth arrest. But Henrietta's cancer cells kept dividing. They doubled every 20 to 24 hours. They were vigorous, resilient, easy to grow. Researchers started sharing them. Labs around the world began using them for experiments. Those cells are still alive today. HeLa cells, they're called, named after Henrietta. They've been reproducing for over 70 years through billions of cell divisions, in labs on every continent. They've been to space. They've contributed to thousands of scientific studies, from polio vaccine development to cancer research to COVID-19 treatments. Henrietta Lacks died in 1951, but her cells are immortal. This is what cancer does. It unlocks the mechanisms of indefinite growth, and in doing so, it reveals something profound about aging. The capacity for immortality is already inside us. Every cell in your body contains the genetic instructions for unlimited division. The machinery is there, it's just turned off. Normal cells are mortal by design. They have limits built in at multiple levels. Telomeres shorten with each division. Tumor suppressor genes activate if something goes wrong, forcing the cell to stop dividing or self-destruct. DNA damage triggers repair mechanisms, or if the damage is too severe, programmed cell death. These are safeguards, layers of protection to prevent any single cell from going rogue and growing out of control. Cancer is what happens when those safeguards fail. A cell accumulates mutations, sometimes from random errors during DNA replication, sometimes from environmental damage like UV radiation or carcinogens, sometimes from inherited genetic vulnerabilities. Most of the time, the mutations are caught and repaired, or the cell is destroyed. But occasionally, a cell accumulates just the right combination of mutations to bypass the safeguards. It reactivates telomerase, the enzyme that rebuilds telomeres, giving itself unlimited division potential. It disables p53, the tumor suppressor gene that normally triggers cell death in damaged cells. It ignores growth signals from neighbouring cells. It starts producing its own growth factors, telling itself to divide without external permission. It becomes autonomous, immortal, and then it starts taking over. According to research on the hallmarks of cancer, published by Hanahan and Weinberg in 2000 and updated in 2011, there are specific capabilities that cells need to acquire to become cancerous. Sustained proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis to build new blood vessels, activating invasion and metastasis. Later updates added avoiding immune destruction and reprogramming energy metabolism. Each of these is a step away from cooperation and towards selfishness. A normal cell exists as part of a community, coordinating with its neighbours, following the rules that keep the whole organism functional. A cancer cell abandons that. It becomes a rebel, optimizing for its own survival and reproduction at the expense of everything else. This is cellular immortality without coordination. Immortality is catastrophe. And here's the paradox. Cancer uses many of the same molecular pathways that Turritopsis uses to achieve its controlled immortality. Telomerase reactivation, stem cell factors like OCTIS4 and SOX2, resistance to apoptosis, programmed cell death. The difference is regulation. Turritopsis activates these pathways in a coordinated way across the entire organism, following a developmental program that rebuilds the body into a functional polyp. Cancer activates them chaotically in individual cells, creating a mass of undifferentiated cells that serve no function except their own replication. It's the same toolkit, applied in two completely different ways. One builds, one destroys. This is why aging exists in the first place, not because immortality is impossible, but because the mechanisms that enable immortality are dangerous. They have to be kept under tight control, or they turn into cancer. Evolution solved this problem by building in aging, by making cells mortal, by ensuring that even if a cell escapes some of the safeguards, it will eventually hit the telomere limit and stop dividing. Aging is, in part, a cancer prevention strategy. According to the antagonistic pleiotropy theory of aging, proposed by George Williams in 1957, many of the genes that cause aging later in life are actually beneficial early in life. They promote growth, enable development, enhance fertility. But those same genes have negative effects later, contributing to cellular senescence, tissue degradation, increased cancer risk. Evolution favours genes that help you survive and reproduce during your reproductive years, even if those same genes harm you after reproduction is done. Because by the time the harm kicks in, you've already passed on your genes. Natural selection can't see beyond the reproductive window. This is why cancer risk increases exponentially with age. The safeguards that prevent cancer, cell cycle checkpoints, DNA repair, immune surveillance, all decline as we get older. Not because they wear out randomly, but because evolution didn't prioritise maintaining them past reproductive age. You've already reproduced. The species has moved on. But from an individual perspective, this is a raw deal. You want to live longer. You don't want to develop cancer. But the very mechanisms that would let you live longer, reactivating telomerase, boosting cell proliferation, enhancing stem cell activity, are the same mechanisms that increase cancer risk. This is the tightrope that longevity research has to walk. How do you rejuvenate tissues without triggering cancer? How do you reactivate the machinery of youth while keeping it under control? Teratopsis has an answer, but we don't fully understand it yet. Part of the answer seems to be timing. The jellyfish only activates its reprogramming during specific stress conditions and only for a limited time. It's not running telomeres constantly. It's not maintaining high levels of pluripotency factors indefinitely. It pulses them, turns them on during transformation, then turns them off once the polyp is established. This is similar to what researchers found in those 2016 experiments with mice. Brief pulsed activation of the Yamanaka factors could rejuvenate tissues without causing cancer, but constant activation led to tumours. The key was dosage and timing. There's also likely something about the jellyfish's tissue architecture that makes the process safer. Jellyfish don't have solid organs with complex structures that can be disrupted. Their bodies are mostly water, held together by a simple epithelial layer and a diffuse nerve net. De-differentiation doesn't destroy critical systems, because there aren't that many critical systems to destroy. Mammals are different. We have organs that can't be turned off, even temporarily. Your heart has to keep beating, your lungs have to keep breathing, your brain has to keep processing. If those cells de-differentiate, even for a short time, you die. So whole body reprogramming isn't an option for us, but tissue-specific reprogramming might be. What if you could rejuvenate your skin without affecting your liver? Reset your muscle cells without touching your neurons? Deliver reprogramming factors locally to specific tissues in controlled doses without systemic effects? This is being explored in labs right now. Researchers are developing ways to target cellular reprogramming to specific organs or cell types, using gene therapy vectors that only activate in certain tissues. Designing drugs that can temporarily induce pluripotency factors and then clear from the system quickly. The goal is to get the benefits of cellular rejuvenation, restored function, reduced senescence, improved repair, without the risks of uncontrolled proliferation. But even with perfect targeting and perfect timing, there's still the cancer problem. Because here's the thing about cancer. It's not just external. It's not just mutations from environmental damage. Some cancer risk is intrinsic to being a complex, long-lived organism. Every time one of your cells divides, there's a small chance of a copying error in the DNA. Most errors are harmless. Some are caught and repaired. But occasionally an error lands in a gene that matters, a tumor suppressor, a growth regulator, and that cell is now one step closer to cancer. This is called the somatic mutation theory of cancer, and it means that cancer risk accumulates over time simply from living. The longer you live, the more cell divisions you undergo, the more chances for something to go wrong. According to research by Tomasetti and Vogelstein, published in Science in 2015, much of the variation in cancer risk between different tissue types can be explained by the number of stem cell divisions those tissues undergo. Tissues that divide frequently, colon, skin, lung, have higher cancer rates. Tissues that divide rarely, brain, heart, have lower rates. It's a numbers game. More divisions, more chances for mutation. If we extend human lifespan significantly, we increase the number of cell divisions each person undergoes, which means more opportunities for cancer to develop, unless we also improve DNA repair, enhance immune surveillance, or find ways to clear precancerous cells before they become dangerous. Some organisms have solved this. Large animals like elephants and whales should have much higher cancer rates than small animals like mice, simply because they have more cells. More cells means more divisions means more chances for cancer. But they don't. Elephants have lower cancer rates than humans, despite having 100 times more cells and living for decades. This is called Peto's paradox, named after epidemiologist Richard Peto, who first noted it in the 1970s, and the solution appears to be that large, long-lived animals evolved extra cancer suppression mechanisms. Elephants, for example, have 20 copies of the 53 gene, the tumor suppressor that triggers cell death in damaged cells. Humans have 1 copy. According to research published in 2015 by Joshua Schiffman and colleagues, those extra copies make elephant cells much more sensitive to DNA damage. They die more readily when something goes wrong, preventing potential cancers before they start. Whales have their own solutions: enhanced DNA repair, more efficient cellular quality control, mechanisms we're only beginning to understand. The point is, cancer resistance can be enhanced. Evolution has done it multiple times in different lineages. The mechanisms exist. We just need to identify them and figure out how to apply them to humans. This is one of the promises of studying Turritopsis. The jellyfish undergoes massive cellular reprogramming without developing cancer. Whatever regulatory networks it's using to keep the process safe, those networks are valuable. They represent millions of years of evolution solving the exact problem we're trying to solve. But there's a darker side to this as well, a possibility that makes some researchers uncomfortable. What if aging is the price we pay for complexity? What if the only way to build a brain capable of consciousness, a body capable of abstract thought and tool use and language, was to accept mortality as the trade-off? What if, in other words, you can't have both? Not with biology as it currently exists. This is speculative, but there's a logic to it. The more complex a system is, the more ways it can fail. The more specialized cells are, the harder they are to repair or replace. The more information is stored in structures, like the connections between neurons, the more dangerous it is to erase those structures. Turritopsis can be immortal because it's simple. It can de-differentiate completely and still survive because there's not much to lose. But we're not simple, we're profoundly complex. And that complexity might be incompatible with the kind of immortality the jellyfish achieves. Maybe the best we can do is extend lifespan incrementally, push the boundaries but not break them. Live to 120 or 150 or 200, but not indefinitely. Keep aging at bay longer but not forever. Or maybe we're missing something. Maybe there's a solution we haven't thought of, a way to preserve complexity while resetting aging that doesn't exist in nature yet but could be engineered. Because we're not bound by evolution anymore. Evolution works through random mutation and natural selection, a slow, blind process that can only build on what already exists. But we have foresight. We have technology. We can design solutions that evolution never explored. This is the promise and the peril of synthetic biology. The idea that we can rewrite the code of life itself, not just to fix diseases but to enhance capabilities, extend lifespan, maybe even achieve immortality through means that nature never invented. But every time we talk about rewriting biology, we have to confront the cancer problem, because cancer is what happens when cells rewrite their own biology in uncontrolled ways. It's a preview of what could go wrong if we're not careful. Turritopsis is showing us that controlled cellular reprogramming is possible, that immortality and organization can coexist. But it's also reminding us that the line between rejuvenation and catastrophe is razor thin, and as we get closer to crossing that line ourselves, we need to decide, Is this a boundary we want to cross? And if we do cross it, how do we make sure we end up like Turritopsis, renewed and functional, and not like cancer, immortal but destructive? Let's say we figure it out. Let's say we map every gene involved in Turritopsis' transformation, identify every regulatory network, understand the complete molecular choreography. Let's say we develop a therapy that can de-differentiate your cells, erase decades of aging damage, and rebuild your tissues into a younger version of yourself. Would you still be you? This isn't a hypothetical question. It's the core philosophical problem that immortality forces us to confront. Because if every cell in your body is replaced, if the physical substrate of your existence is renewed, what happens to the person who inhabited that substrate? Your body replaces itself constantly anyway. Skin cells turn over every few weeks. Red blood cells last about 4 months. Most of your skeleton is replaced every 10 years. The atoms that make up your body today are not the atoms that made up your body 7 years ago. You're already a different physical entity than you were in the past. But there's one part of you that doesn't replace itself. Your neurons. Most of the neurons in your brain were formed before you were born, and they stay with you for life. They grow, they change, they form new connections and prune old ones, but the cells themselves persist. Your memories are encoded in the pattern of connections between those neurons. Your personality, your skills, your sense of self, all of it is physical structure in your brain. If you de-differentiate your neurons, even temporarily, that structure is erased. The connections are lost, the information is gone. You could rebuild a brain. You could re-differentiate the cells back into neurons, form new connections, establish new patterns, but it wouldn't be the same brain. The memories wouldn't return. The person who woke up would be someone new, wearing your face, living in your body, but fundamentally different. This is what happens to Turritopsis. When the medusa transforms into a polyp, it loses everything. The neural net that coordinated its swimming, its feeding, its responses to stimuli, all of that is broken down and rebuilt from scratch. The polyp that emerges is genetically identical to the medusa, but it's not the same individual in any meaningful sense. It has no memory of being a medusa. It has no continuity of experience. Turritopsis achieves immortality by dying and being reborn, over and over. The organism persists, but the individual does not. For humans, this might not be acceptable. We don't just want our bodies to continue, we want ourselves to continue, the unbroken thread of consciousness and memory that makes us who we are. We want to live longer, not to be replaced by younger copies of ourselves. This is the ship of Theseus problem, one of the oldest thought experiments in philosophy. If you replace every plank in a ship, one at a time, until none of the original material remains, is it still the same ship? If you collect all the old planks and build a second ship from them, which one is the real ship of Theseus? Most people's intuition is that the ship that was continuously repaired, piece by piece, is the real one, because there was never a moment when it stopped being the ship. Identity persists through gradual change, even if the material substrate is completely replaced. But what if the change isn't gradual? What if you disassemble the entire ship at once, build it back from scratch using new materials, following the same blueprint? Is that the same ship, or is it a replica? Your body does the gradual version naturally. Cells are replaced slowly, over time, while you remain conscious and aware throughout the process. You experience continuity. You remember yesterday, and the day before, and your childhood. The thread of self remains unbroken even as the physical substrate changes. But Turritopsis' method isn't gradual, it's catastrophic transformation. The medusa collapses, the polyp emerges, there's no continuity of consciousness because there's no consciousness to maintain. Jellyfish don't have subjective experience in the way we do. They're reflex machines, nerve nets that respond to stimuli but don't think, don't remember, don't have an inner life. For them, this doesn't matter. The organism survives. That's enough. But we're different. We have inner lives. We have memories we want to keep, relationships we want to preserve, skills we've spent years developing. We are not just biological machines. We are experiencing subjects. And any immortality treatment that erases experience is not immortality. It's death with extra steps. This is why partial reprogramming is so important. If we can rejuvenate tissues without de-differentiating neurons, we might be able to extend lifespan while preserving self. Reset the body but not the mind. Keep the hardware running while maintaining the software. But even this gets complicated. Because your brain isn't separate from your body. It's connected to every system, constantly receiving signals, adjusting based on hormones and metabolic states and gut bacteria, and dozens of other inputs. Change the body significantly, and you change the brain's environment, which might change how it functions, which might change who you are. According to research in neuroscience and psychology, personality isn't fixed. It changes throughout life in response to experiences, relationships, physical health, even diet. The person you are at 20 is not the person you are at 50. Even if your memories remain continuous, we're constantly being reshaped by the feedback loops between our bodies, our environments, and our minds. If a rejuvenation treatment resets your body to a biologically younger state, would it change your personality? Would a 70-year-old with the hormones and metabolism of a 25-year-old think and feel like a 25-year-old? Or would they retain the psychological traits and wisdom accumulated over decades? We don't know, but it matters. Because if rejuvenation erases psychological maturity, it's not clear that it's a good thing. You might live longer chronologically, but if your mind regresses to an earlier state, have you really gained time? Or have you just looped back to an earlier save point, losing everything you learned along the way? These questions don't have clear answers, and they become even more complex when you consider memory itself. Memory isn't stored in neurons the way data is stored on a hard drive. It's not localized to specific cells that could be protected while other cells are rejuvenated. Memory is distributed across networks of neurons, encoded in the strength and pattern of synaptic connections. It's the physical structure of how your neurons link together. According to research on memory consolidation, long-term memories are stored through physical changes to synapses, the junctions between neurons where signals pass from one cell to another. When you learn something new, repeated activation of a neural pathway strengthens the synapses along that path. The connections become more efficient, more likely to fire in the future. That's memory. It's literally the shape of your brain's wiring. If you de-differentiate neurons, you erase that wiring. The synapses are lost. The memories go with them. Some researchers have proposed that memory might be encoded in other ways as well, in epigenetic markers on DNA, in prion-like proteins that can propagate states between cells, in non-neuronal cells like glia that support neurons. If that's true, there might be ways to preserve memory even while replacing neurons. Encode the information in a substrate that survives de-differentiation, then restore it when new neurons form. But this is highly speculative. We don't have evidence that memory can survive the kind of transformation teratopsis undergoes. And even if it could, there's the question of whether a restored memory is the same as an original memory. If you write down everything you remember, erase your brain, and then read your notes and reconstruct the memories from scratch, are those the same memories? Or are they copies? Does it matter? Some philosophers argue that identity isn't about physical continuity at all. It's about psychological continuity. As long as your memories and personality traits are preserved, you're the same person, even if the physical substrate changes completely. This is called the psychological continuity theory of personal identity. By this logic, if we could back up your memories and personality, record every synapse, every connection, every pattern, and then restore them after rejuvenation, you would still be you. The break in physical continuity wouldn't matter because the psychological continuity would be maintained. But this assumes that identity is information, not matter, that you are the pattern, not the stuff that embodies the pattern. And while that's appealing philosophically, it's deeply counterintuitive. Most people feel that they are their bodies, not just data that could be transferred between substrates. There's also a practical problem. We don't know how to back up a human mind. The technology to record every synapse, every connection, every relevant state in a living brain doesn't exist. And even if it did, the resolution required would be staggering. The human brain contains roughly 86 billion neurons, each connected to thousands of others. Mapping all of that in sufficient detail to reconstruct a mind would require imaging technology and computational power far beyond what we currently have. So for the foreseeable future, any rejuvenation treatment that affects the brain will be a gamble. We won't know if it preserves identity until after it's done, and if it doesn't, the person who agreed to the treatment won't be around to regret it. This is one reason why early longevity research is focusing on peripheral tissues, skin, muscle, liver, heart, and avoiding the brain entirely. Those tissues can be rejuvenated without threatening identity. You can have a younger body and still be yourself. Your memories remain. Your personality persists. But there's a limit to how much this approach can achieve, because the brain ages too. Neurons accumulate damage, connections are lost, cognitive decline sets in. If you rejuvenate everything except the brain, you end up with a young body and an old mind. That might extend lifespan, but it won't prevent dementia, won't preserve cognitive function, won't keep you mentally sharp as the decades pass. At some point, if we want true longevity, we have to address the brain, and that means confronting the identity problem head-on. Maybe the answer is to accept imperfect continuity, to acknowledge that the person who undergoes brain rejuvenation will be slightly different afterward. Some memories lost, some personality traits shifted, but still recognisably themselves, still continuous enough to count as the same person. This is how we already live after all. You're not identical to who you were 10 years ago. Memories fade, perspectives change, personality evolves, but there's enough continuity that you still consider yourself the same person. You're the ship of Theseus, with planks replaced gradually over time. Maybe rejuvenation would just accelerate that process. More change, faster, but still within the bounds of acceptable continuity. Or maybe we need to rethink what we mean by immortality. Maybe true immortality, eternal life as the same unchanging self, is neither possible nor desirable. Maybe what we should be aiming for is extended life with meaningful continuity, not freezing ourselves in time, but slowing the rate of change, living longer, accumulating more experiences, but still growing and evolving as people. Turritopsis doesn't offer this. It offers reset, not continuation. But Turritopsis is a jellyfish. It has no self to preserve. We do. And that makes our problem fundamentally different. The jellyfish can cycle through life stages indefinitely because it has nothing to lose in the transition. We have everything to lose. Our memories, our relationships, our accumulated wisdom, the person we've become through decades of living. Any immortality treatment for humans has to preserve that, or at least preserve enough of it that we recognize ourselves on the other side. This is the challenge that makes human immortality so much harder than jellyfish immortality. It's not just a question of biology, it's a question of identity, consciousness, and what it means to be a person extended across time. And there's one more layer to this problem that we haven't addressed yet, one that might be the most difficult of all. Even if we solve the biology, even if we figure out how to rejuvenate the body while preserving the mind, even if we achieve true human immortality with perfect continuity of self, would we want it? Would living forever actually be a good thing? For individuals? For society? For the species? Because Turritopsis might have solved the technical problem of immortality, but it hasn't had to grapple with what immortality means. It just lives, cycles, continues, without reflection or consequence. We can't do that. We think, we plan, we consider the future. And when you imagine a future without end, the questions get uncomfortable very quickly. There's a thought experiment that philosophers have debated for centuries. Imagine you're offered immortality, true immortality. You will never age, never die from disease, never cease to exist. You can still be killed by accident or violence, but otherwise you'll live as long as the universe permits. Do you take it? Most people's immediate answer is yes. Of course. Who wouldn't want to live forever? Death is the ultimate loss, the end of everything you are and everything you could become. Immortality means infinite time to learn, to explore, to experience, to connect with others. It's the ultimate gift. But then you start thinking about what forever actually means, and the answer gets more complicated. Forever isn't just a long time, it's all time. Every possible experience repeated infinitely. Every conversation eventually exhausted. Every place eventually visited. Every sensation eventually dulled by familiarity. In a finite life, novelty is guaranteed. You'll die before you run out of new things to experience. But in an infinite life, novelty becomes impossible. You'll eventually do everything, see everything, exhaust every possibility. And then what? You keep living, but there's nothing new, only repetition, variation on themes you've already explored, echoes of experiences you've already had. This is the paradox of immortality. It promises infinite possibility but guarantees eventual meaninglessness, because meaning often comes from scarcity, from the fact that time is limited, choices matter, opportunities are finite. When you have forever, nothing is urgent, nothing is precious. There's always more time. The philosopher Bernard Williams explored this in a famous essay called The Microscopic Case, named after a play where a woman becomes immortal at age 42 through an alchemical elixir. After 300 years, she's deeply depressed, bored beyond measure, indifferent to life. She chooses to let the immortality expire and die, because eternal life has become unbearable. Williams argued that immortality would necessarily lead to this state. Either you remain the same person forever, in which case you eventually exhaust everything that interests you and become bored, or you change continuously to remain engaged, in which case you eventually become someone so different that the original you has effectively died anyway. It's a lose-lose scenario. Immortality either means eternal stagnation or gradual erasure of self. But this argument assumes that meaning and engagement require finitude, that we need death to give life value. And that's not obviously true. You could argue that humans adapt to almost anything. We adapt to wealth, to poverty, to disability, to chronic illness. Our baseline happiness tends to return to a set point regardless of external circumstances. Maybe we'd adapt to immortality too. Maybe after a few centuries the novelty would wear off and we'd settle into a new equilibrium. Life would feel normal again, just longer. Or maybe the problem isn't immortality itself, but the assumption that you'd remain isolated. Williams's example is a single immortal person living among mortals. Of course, that would be lonely. Everyone you love dies. Every relationship ends. You're perpetually losing people, accumulating grief, unable to form lasting connections because you outlive everyone. But what if everyone was immortal? What if we're not talking about one person living forever, but a whole civilization that never dies? That changes the dynamics entirely. Relationships wouldn't end from death. Knowledge wouldn't be lost between generations. People could specialize for centuries, becoming true experts in narrow fields. Culture could develop in ways impossible when lifespans are short. But it also creates new problems. Social problems, economic problems, political problems. Start with population. If people stop dying but keep reproducing, population grows exponentially. Within a few generations, Earth would be overcrowded beyond sustainability. Resources would be depleted. Living space would become scarce. We'd be forced to choose between immortality and having children. Most societies would probably choose to restrict reproduction. No new people, or very few, so the existing population can persist indefinitely. But that creates a static society. No new perspectives, no generational turnover. The same people, holding the same positions, making the same decisions forever. Innovation tends to come from youth, from people who haven't yet learned that something is impossible or haven't yet internalized the existing paradigm. Einstein developed relativity in his 20s. Most mathematicians do their best work before age 40. Revolutionary ideas often come from outsiders, from people who see problems with fresh eyes. In an immortal society, there are no fresh eyes. Everyone has centuries of experience, centuries of accumulated assumptions. The older you get, the harder it becomes to change your mind, to adopt new frameworks, to see things differently. Immortality might mean not just individual stagnation, but cultural stagnation. According to research on cognitive aging, while older adults accumulate knowledge and expertise, they tend to become less cognitively flexible. Crystallized intelligence, the ability to use learned knowledge, increases with age. But fluid intelligence, the ability to think abstractly, solve novel problems, adapt to new situations, declines. This isn't just about neurons dying, it's about the brain becoming optimized for existing patterns at the expense of novelty. If we extend lifespan indefinitely without addressing this, we might end up with a society of rigid thinkers, unable to adapt to changing circumstances, locked into patterns established centuries ago. And then there's power. In human societies, power tends to accumulate. Wealth compounds, influence grows. The longer someone lives, the more resources they can amass, The more connections they can establish, the more control they can exert. Now imagine that person lives not 80 years, but 800 or 8,000. The concentration of power would be staggering. Billionaires who never die, politicians who never leave office, CEOs who run corporations for centuries. The established order would become truly permanent. Younger generations, if they exist at all, would have no chance to rise. Every position would be occupied by someone with centuries of experience. Every opportunity would be claimed by someone who's been around longer. Inequality would solidify into something immovable. This is the dark side of immortality that science fiction often explores. A gerontocracy, ruled by the old, that never ends. A society where change becomes impossible because the people in power never die and never relinquish control. There are potential solutions: term limits, forced retirement, wealth redistribution. Mechanisms to ensure that immortality doesn't mean permanent entrenchment, but implementing those mechanisms requires governance structures that we haven't built yet, and it requires immortal people to agree to give up power, which seems unlikely. Then there's the question of meaning at a personal level. Not philosophical boredom, but practical purpose. What do you do with your time when you have infinite time? For most of human history, survival itself consumed most of our energy. Finding food, avoiding predators, staying warm, raising children. Purpose was built into the struggle to persist. But in modern developed societies, survival is relatively easy. We've automated much of the labour. We have time to ask, what's the point? And we fill that time with goals, career advancement, creative projects, learning new skills, building relationships, raising children. These goals give structure to life. They provide milestones, achievements, a sense of progress. But most of these goals are implicitly tied to mortality. You build a career knowing you have 40 or 50 years to work, then retirement. You raise children knowing you're preparing them for a world you won't always be part of. You pursue creative projects knowing they're part of your legacy, something that will outlast you. Remove mortality, and the urgency vanishes. Why finish that novel this year when you have infinite years to finish it? Why change careers now when you can always do it later? Why have difficult conversations today when there's always tomorrow? Immortality might lead to perpetual procrastination. Infinite time creates the illusion that nothing needs to be done now. Everything can wait. And if everything can wait, nothing gets done. Some researchers in psychology have studied this. They've found that people facing terminal illness often report increased life satisfaction and clarity of purpose. Not in spite of their limited time, but because of it. Knowing that life is short makes people focus on what truly matters. They stop putting things off. They repair relationships. They pursue meaningful goals. Mortality creates urgency. Without it, we might drift. Not unhappy, necessarily, but aimless. Floating through centuries without direction or purpose, because there's no deadline to create focus. And then there's the problem of memory. Even if we solve the technical challenge of preserving memories during rejuvenation, there's a limit to how much a human brain can store. According to estimates from neuroscience, the human brain has a storage capacity somewhere between 1 and 10 terabytes. That's enormous, but it's finite. If you live for 80 years, you'll never fill that capacity. But if you live for 800, at some point, you run out of room. Old memories have to be overwritten to make space for new ones. You start forgetting your past. Centuries of experience fade, become inaccessible. At that point, are you still the same person? If you've lived for 1,000 years but can only remember the last 200, what happened to the other 800 years of you? Did that person die? Or are they still you, just lost to time? This is another version of the identity problem, but stretched across a timescale that makes it existential. You become a being that's constantly losing itself, shedding old selves to make room for new ones, like a snake shedding skin. Continuous, but not constant. Persistent, but not permanent. Maybe that's acceptable. Maybe that's even desirable. You get to reinvent yourself repeatedly, exploring different versions of who you could be, without the constraint of a single static identity. Or maybe it's a kind of death. Slow, gradual, invisible, but still death. The person you were at age 100 is gone by age 1000. They were replaced by someone new, wearing the same body, carrying a few remnants of memory, but fundamentally transformed. Turritopsis doesn't worry about any of this. It has no memory to preserve, no identity to maintain, no sense of purpose beyond survival. It cycles through transformations without self-awareness, without questioning, without suffering the existential weight of infinite time. But we would. We can't not think about it. And that thinking might be the hardest part of immortality. Because immortality isn't just a biological challenge, it's a psychological one, a social one, a philosophical one. Even if we solve the science, even if we map every pathway, understand every mechanism, develop treatments that can safely rejuvenate tissues while preserving identity, we'd still have to answer the harder questions. Do we want this? Should we pursue it? And if we achieve it, how do we live with it? These aren't questions that Turritopsis can help us answer. The jellyfish just exists, immortal and indifferent. It's up to us to decide what immortality would mean for beings who are conscious, reflective, burdened with the knowledge of their own existence. And maybe the answer is that we shouldn't aim for true immortality. Maybe the goal should be something less absolute. Extended lifespan, yes. Health span that matches lifespan, absolutely. Rejuvenation therapies that allow people to remain vital and engage for longer, but not forever. Maybe 150 years is enough, or 200. Long enough to see your great-great-grandchildren grow up. Long enough to master multiple fields, explore different careers, live several complete lives within one lifespan, but still finite, still bounded by death, which gives shape to life. This is the middle path that some longevity researchers advocate for, not defeating death, but postponing it. Not breaking the limits entirely, but pushing them outward. Giving people more time, but not infinite time. And maybe that's wise. Because the more we think about what true immortality would mean, the less appealing it becomes. Not because of the biology, Turritopsis proves the biology is possible, but because of what it would do to us as thinking, feeling, meaning-making beings. Still, the research continues. The mechanisms are being mapped, the pathways are being understood, and regardless of whether we ever achieve or even want true immortality, the knowledge we're gaining has immediate applications. Because understanding how Turritopsis reverses aging teaches us about aging itself, about why it happens, about how it could be slowed or partially reversed, about the trade-offs between longevity and other biological functions, and that knowledge doesn't just apply to immortality, it applies to age-related diseases, to degenerative conditions, to the physical and cognitive decline that affects almost everyone who lives long enough. Even if we never make anyone immortal, we might make the time people do have better, healthier, more functional, less burdened by the breakdown that characterizes old age. That's a goal worth pursuing, not forever, but better. Not immortality, but vitality. And Turritopsis, drifting in the ocean, indifferent to philosophy and ethics and the weight of infinite time, is showing us how. While philosophers debate the meaning of immortality, researchers are working in labs around the world trying to translate what Turritopsis does into something applicable to humans. Not immortality, necessarily, but some version of cellular rejuvenation that could extend health span, reverse aspects of aging, or repair tissues that have degraded over time. The work is painstaking. Jellyfish are not easy experimental subjects. They're fragile, difficult to culture in large numbers, sensitive to water quality and temperature. Turritopsis dorani specifically is tiny. Adults are only a few millimeters across. Working with them requires specialized equipment, patience, and a tolerance for setbacks, but the potential payoff is enormous. So researchers persist. One of the key questions is, which genes are essential? During transdifferentiation, thousands of genes change their expression levels. Some are turned on, some are turned off, some are modulated up or down. Figuring out which changes are critical and which are just side effects is like trying to understand a machine by watching all its parts move at once. You can see that gears are turning, pistons are firing, but it's not immediately obvious which movements cause which outcomes. To untangle this, researchers use comparative genomics. They sequence the genomes of multiple jellyfish species, some that can transdifferentiate, some that can't, and look for differences. Which genes does Turritopsis have that close relatives lack? Which regulatory regions are unique? What evolutionary changes enabled this ability? According to research published in 2022 by a team at the University of Oviedo led by Maria Pascal Torner, Turritopsis has specific genetic signatures that distinguish it from non-transdifferentiating species. Genes related to DNA repair are more active. Genes involved in maintaining pluripotency, the ability of cells to become multiple types, show unusual patterns of regulation. There are also differences in how the jellyfish manages oxidative stress, the cellular damage caused by reactive oxygen species that accumulates during normal metabolism. One finding stands out. Turritopsis appears to have enhanced protection against oxidative damage during transdifferentiation. This makes sense. When cells dedifferentiate and then redifferentiate, they're under enormous metabolic stress. They're breaking down and rebuilding structures, which generates a lot of cellular waste and reactive molecules. If that damage isn't managed carefully, it can lead to mutations, cell death, or cancer. The jellyfish has evolved mechanisms to handle this stress. Antioxidant systems that neutralize reactive molecules, DNA repair pathways that fix damage quickly, quality control systems that identify and destroy defective proteins before they cause problems. These aren't unique to Turritopsis. All animals have versions of these systems, but in the immortal jellyfish, they seem to be particularly robust, tuned to handle the extreme stress of transformation. This is useful information for human applications, because one of the major challenges in cellular reprogramming is managing the stress it creates. When you introduce Yamanaka factors into adult cells to turn them into stem cells, the process is inefficient and often results in cell death or genomic instability. Most cells don't survive. The ones that do sometimes have mutations. If we understood exactly how Turritopsis protects its genome during dedifferentiation, we might be able to apply those same protective mechanisms to human cells during reprogramming. Make the process safer, more efficient, less likely to cause problems downstream. Some labs are already testing this. They're taking human cells, introducing reprogramming factors, and simultaneously boosting antioxidant systems or DNA repair pathways. Early results suggest it helps. The cells reprogram more efficiently, fewer die in the process. The resulting stem cells are healthier, with fewer genetic abnormalities. This is incremental progress, but it's progress. Small steps toward making cellular rejuvenation safe enough for clinical use. Another area of focus is tissue-specific reprogramming. The idea is that instead of trying to rejuvenate the entire body at once, which would be incredibly dangerous, you target specific tissues or organs that are aging or damaged. For example, the heart. As you age, cardiac cells accumulate damage. They become less efficient at contracting, less responsive to signals. This contributes to heart disease, the leading cause of death worldwide. But cardiac cells are postmitotic. They don't divide after early development. Once they're damaged, they don't regenerate. What if you could trigger partial reprogramming in cardiac tissue? Not full dedifferentiation, which would stop the heart from beating, but just enough to reset some aging markers and restore function. Reset the cell's clocks without erasing their identity. This has been attempted in mice. In 2020, researchers at the Salk Institute published a study where they delivered reprogramming factors specifically to heart tissue in aged mice. The treatment was brief, just long enough to induce some rejuvenation, without causing the cells to lose their identity as heart cells. The results were promising. The treated hearts showed improved function. Cells were metabolically younger. Some markers of aging were reversed, and critically, the mice didn't develop tumours. The reprogramming was controlled enough to be safe. This is the kind of application that might reach humans within decades. Not immortality, not even radical life extension. Just targeted repair of aging tissues, one organ at a time, extending health span by keeping critical systems functional longer. The same approach is being explored for other tissues. Muscle, which weakens with age. Liver, which loses regenerative capacity. Skin, which thins and loses elasticity. Bone marrow, where stem cells become depleted. Each tissue presents different challenges, but the underlying principle is the same. Use the lessons from Turritopsis and other regenerative organisms to develop safe, controlled rejuvenation therapies. There's also work on senescent cells that have stopped dividing but haven't died. They accumulate with age, and they secrete inflammatory molecules that damage surrounding tissues. Clearing these cells in mice extends lifespan and improves health. Drugs called senolytics, which selectively kill senescent cells, are now in human clinical trials. This is another lesson from studying immortal and long-lived organisms. Turritopsis doesn't accumulate senescent cells during its transformation. It either repairs them or destroys them, clearing the slate before rebuilding. We can learn from that, develop therapies that do the same in human tissues. Then there's the question of the immune system. As you age, your immune system declines. T cells become exhausted. The thymus, where new T cells are produced, shrinks. You become more vulnerable to infections, less able to fight cancer, less responsive to vaccines. But the thymus isn't permanently dead, it's just dormant. Research in mice has shown that you can rejuvenate the thymus using growth factors or partial reprogramming. The organ regrows, new T cells are produced, the aged immune system regains function. This has profound implications for aging and disease. A rejuvenated immune system could prevent infections, clear precancerous cells, reduce chronic inflammation that drives many age-related diseases. It's one of the most promising targets for near-term rejuvenation therapies. Again, the inspiration comes from organisms like Turritopsis. When the jellyfish transforms, it rebuilds its entire body, including immune components. It doesn't carry forward the accumulated dysfunction of adulthood. It resets. We can't reset as completely as a jellyfish can, but we might be able to reset partially. Target the systems that matter most for health span: immune, cardiovascular, musculoskeletal, nervous, and rejuvenate them selectively. This is the pragmatic path forward. Not chasing immortality as an endpoint, but using the biology of immortal organisms to develop practical therapies that improve human health. And the research is accelerating. According to data from the longevity biotech sector, investment in aging research has increased dramatically over the past decade. Dozens of companies are now working on senolytic drugs, partial reprogramming therapies, immune rejuvenation, metabolic interventions. Clinical trials are underway for multiple approaches. Some of these will fail, most, probably, but some will succeed. And even modest success, extending healthy lifespan by 5 or 10 years, would have enormous impact. Delaying the onset of age-related diseases, keeping people functional and independent longer, reducing the burden of chronic illness. That's the realistic goal. Not defeating death, but postponing decline. But even as this work progresses, researchers studying Turritopsis keep uncovering new mysteries. Because the more we learn about the jellyfish, the more we realize how much we don't understand. For instance, how does the jellyfish know when to transform? What signals trigger the decision? It's not automatic. The jellyfish doesn't reverse every time it's stressed. Sometimes it just dies, like any other animal. So there's a decision point, a threshold where the organism commits to transformation. What determines that threshold? Is it the severity of damage, the availability of energy reserves, some combination of internal and external factors? And how is that information integrated across the entire organism to produce a coordinated response? We don't know yet. We see the transformation happen, but we don't fully understand the logic that governs it. There's also the question of limits. Can Turritopsis cycle indefinitely, truly forever, or is there an upper limit? In the lab, researchers have documented dozens of cycles in individual jellyfish, but no one has tracked a single individual for centuries. Maybe after 1,000 cycles, something breaks down. Maybe there's an accumulation of subtle errors that eventually becomes catastrophic, or maybe not. Maybe the process really is perfect, and the jellyfish genuinely never has to die. Testing this would require decades of observation, maintaining cultures, tracking individuals, documenting every cycle. It's not glamorous work, and it doesn't yield quick results. But it's essential if we want to understand the true limits, or lack thereof, of biological immortality. And then there's the ecological question. In the wild, Turritopsis is not particularly abundant. It exists, it survives, but it's not dominant. If immortality is such a powerful advantage, why hasn't it taken over? Why aren't the oceans filled with immortal jellyfish, outcompeting everything else? Part of the answer is predation. Immortality doesn't protect you from being eaten. Fish, sea turtles, other jellyfish, all of them consume Turritopsis. The Medusa stage is vulnerable, drifting in open water, easy prey. And the polyp stage, while more protected, is sessile. It it can't move if conditions become unfavourable, if the substrate it's attached to is disturbed, if water quality changes, if predators find it, the polyp dies. Immortality only helps if you can avoid external mortality. There's also competition. Turritopsis isn't the only organism trying to survive and reproduce in its environment. It competes with other jellyfish, with planktonic organisms, with anything that feeds on the same resources. And in that competition, immortality might not be a decisive advantage. Faster reproduction might matter more, or better predation strategies, or tolerance for wider environmental ranges. Immortality is one trait among many. Evolution doesn't optimize for any single trait. It optimizes for reproductive success in specific contexts. And in Turritopsis's context, immortality helps, but it doesn't dominate. This is an important lesson. Even if we develop human rejuvenation therapies, they won't magically solve all problems. People will still get sick. Accidents will still happen. Social and economic challenges won't disappear just because individuals live longer. Immortality, or even extended longevity, is one piece of a much larger puzzle. But it's a piece worth pursuing, because aging causes suffering. Watching your body decline, losing abilities you once took for granted, seeing friends and loved ones deteriorate, it's painful. And much of that pain is treatable, at least in principle. We know aging is not inevitable. Turritopsis proves it. Other organisms prove it in different ways. The biology exists, the mechanisms are there. We just need to understand them well enough to apply them, and we're getting closer. Every genomic study, every experiment in mice, every clinical trial in humans brings us incrementally closer to therapies that could meaningfully extend health span. It won't happen overnight. The complexity is staggering. But the trajectory is clear. We're learning the language of aging, and once we understand it fully, we'll be able to speak back. There's a moment in every documentary about immortality where the narrator zooms out, steps back from the mechanisms and molecules, from the labs and data, and asks the bigger question: what does this mean? We've spent thousands of words following Turritopsis dohrnii through its impossible transformation. We've traced the discovery, mapped the mechanisms, explored the implications for human longevity, wrestled with questions of identity and meaning. But we haven't yet asked the simplest question: what is this jellyfish actually teaching us? Not about telomeres or transcription factors or cellular reprogramming, though those lessons matter, but about life itself, about what it means to exist, to persist, to change while remaining somehow continuous. Because here's what strikes me most about Turritopsis: it survives by letting go. When the medusa faces death, injury, starvation, stress that would kill it, it doesn't fight to maintain what it is. It doesn't cling to its adult form, doesn't try to preserve the body it built, the structures it developed. It releases all of that. It collapses inward, breaks itself down to raw material, and rebuilds from scratch. The organism persists, but the individual form does not. The medusa dies. The polyp that emerges is genetically identical, but experientially new. It has no memory of swimming in open water, no continuity of consciousness, no thread connecting it to what came before except the unbroken chain of cellular division. Turritopsis achieves immortality through radical transformation, by becoming something else. For humans, this feels wrong. We want to persist as ourselves. We want continuity, memory, identity that endures. We're terrified of losing who we are, even if the alternative is death. Better to die as yourself than to live as someone else. But maybe that's our limitation, not a universal truth. Maybe the jellyfish is teaching us that identity is less important than we think, that the self we cling to, the collection of memories and personality traits and preferences that we call me, is just one configuration of matter and energy, useful, meaningful to us, but not sacred, not permanent. You're not the same person you were 10 years ago. The cells have turned over, the memories have faded or been rewritten, the preferences have changed. You've accumulated new experiences, discarded old beliefs, grown in ways that make the past you almost a stranger. And yet you call yourself by the same name. You claim continuity with that past person, even though almost nothing physical remains the same. You're the ship of Theseus, rebuilt plank by plank, insisting you're still the same ship. Teratopsis doesn't pretend, it acknowledges the transformation. It lets the old form die and makes no claim that the new form is the same individual. It just continues. And maybe that's more honest. This doesn't mean we should abandon identity. We're not jellyfish. We have selves that matter, relationships that depend on continuity, social structures built around persistent identity. You can't just decide to become someone else and expect no consequences. But maybe we can learn something from the jellyfish's radical acceptance of change. Maybe we can hold our identities a little more lightly. Recognize that who we are is always in flux, always being rebuilt, and that clinging too tightly to any fixed version of ourselves is a kind of death too. A refusal to grow, to adapt, to let old forms dissolve when they're no longer serving us. There's another lesson here, one about time. We experience time linearly. Born, grow, age, die. It's a one-way arrow, and we're carried along whether we want to be or not. The past recedes, the future approaches. We can't go back, except Teratopsis can. Or at least, it can do something that looks like going back. It resets its developmental clock, returns to an earlier life stage, starts the sequence over. It's not time travel. The jellyfish can't recover the past or revisit earlier moments. But it demonstrates that development isn't as linear as we thought. The arrow can bend. The stages we think of as irreversible, childhood, adolescence, adulthood, old age, are just configurations, patterns that cells have learned to follow, and patterns can be rewritten. This is both hopeful and unsettling. Hopeful because it means biological aging isn't destiny. The trajectory can be altered, damage can be repaired, decline can be reversed, at least partially. Unsettling because it undermines our assumptions about what life is. We think of organisms as moving through time in one direction, accumulating experiences, becoming more complex and differentiated, until eventually the complexity collapses and death occurs. But Teratopsis shows that complexity can be voluntarily reduced. Differentiation can be reversed. The trajectory can loop back on itself. Life is less like a line and more like a cycle, or a spiral, or maybe some shape we don't have words for yet. And if that's true for jellyfish, what does it mean for us? Are we also capable of reversing, of returning to earlier states if only we knew how? Is aging just a program we're running because we haven't learned to edit the code? The evidence suggests yes. The Yamanaka factors prove that adult cells can be reverted to embryonic states. Partial reprogramming experiments show that aging markers can be reset. The machinery for reversal exists in us, even if we don't normally use it. We're not as different from Teratopsis as we like to think. We're just running different software on similar hardware. And maybe that's the deepest lesson. That life is more flexible than it appears. That the constraints we take for granted, aging, death, the forward march of time through a body, are not laws of physics but biological choices. Choices made by evolution, encoded in genes but ultimately changeable. Evolution built aging into us for reasons that made sense in the environment we evolved in. But we're not in that environment anymore. We've changed the world around us. We've eliminated most predators. We've extended childhood. We've built societies where people remain productive and valuable far past reproductive age. The evolutionary logic that made aging adaptive no longer applies, but we're still running the old program, still aging and dying on a schedule that was optimized for a world that no longer exists. Teratopsis never had that problem. It evolved in an environment where the ability to reset was valuable, so it kept the machinery functional. It never turned off the differentiation program. It refined it, optimized it, made it robust enough to use repeatedly. We turned ours off, or rather evolution turned it off for us, because running it in adults was dangerous and offered no reproductive benefit. But the machinery is still there, dormant in our genome, waiting to be reactivated if we can figure out how to do it safely. This is what the jellyfish teaches us ultimately, that the impossible is often just the untried, that biological rules are more like guidelines, that the limits we think are absolute are sometimes just defaults we haven't learned to override. But there's a warning here too, a reminder that every capability comes with risks. Teratopsis's immortality isn't free. It works because the jellyfish is simple, because it can afford to break itself down and rebuild without losing anything critical, because it has no brain to preserve, no complex structures that can't be safely de-differentiated. We're not simple, we're staggeringly complex. And every layer of complexity is a new point of failure, a new way that transformation could go wrong. The same mechanisms that could rejuvenate us could also give us cancer. The same plasticity that could extend our lives could erase our minds. The same flexibility that makes transformation possible makes control difficult. Teratopsis doesn't have to worry about this. It's been doing this for millions of years. The process is refined, tested, debugged by countless generations. The risks have been engineered out. We're just starting. We're trying to learn in decades what took evolution millennia to perfect. And we're trying to apply it to organisms far more complex than jellyfish, with far more ways for things to go wrong. So yes, Teratopsis teaches us that immortality is possible, but it also teaches us humility. The gap between possible and practical is vast. The jellyfish makes it look easy because evolution did the hard work. We're starting from scratch, and maybe that's okay. Maybe we don't need to match what Teratopsis does. Maybe we just need to learn enough to make our finite lives better. Longer, healthier, less burdened by decline. Because here's the final lesson, the one that's easiest to miss. Teratopsis isn't trying to be immortal, it's trying to survive. Immortality is just the strategy it evolved to maximize survival in its particular niche. For us, survival might mean something different. Not living forever, but living well. Not escaping death entirely, but postponing it long enough to see our grandchildren grow up. To finish the projects that matter to us. To experience the things we've always wanted to experience. That's not immortality, but it's enough. And if studying an obscure jellyfish that most people have never heard of brings us closer to that goal, if mapping its genes and understanding its mechanisms gives us tools to extend health span, repair aging tissues, prevent degenerative diseases, then the time spent watching Teratopsis transform in laboratory, aquariums will have been worth it. Not because we'll become immortal, but because we'll understand ourselves better. We'll see that the boundaries of biology are wider than we thought, that aging is not destiny, that the future of human health doesn't have to look like the past. The jellyfish isn't a blueprint for human immortality, it's a proof of concept, a demonstration that mortality is negotiable, that aging can be reversed, that the trajectory of life is more flexible than we assumed. What we do with that knowledge is up to us. We can chase immortality, with all the philosophical and practical complications that entails, or we can aim for something more modest, a longer health span, a better old age, a few more decades of vitality and function. Either way, Teratopsis has shown us the door. Whether we walk through it and where that door leads remains to be seen. But we're looking now, we're paying attention, and that small, translucent jellyfish drifting in darkness, glowing faintly with bioluminescent light, has become a teacher. Not because it knows anything, it's just an animal, following genetic programs, responding to stimuli, surviving. But in its survival, it's revealed something profound about the nature of life itself. That the rules we thought were fixed are actually flexible. That the limits we thought were absolute are actually permeable. That the impossible is sometimes just waiting for someone to figure out how. And maybe that's the real secret to immortality. Not living forever, but living in a way that makes the time you have matter. Learning, growing, changing, letting go of what no longer serves you, and rebuilding from what remains. The jellyfish does this at a cellular level. Maybe we can do it at a psychological one. Maybe true immortality isn't about the body at all, but about how we hold ourselves in time, how we adapt, how we transform, how we persist through change. Teratopsis has been practicing this for millions of years. We're just beginning to understand it, but we're learning. And in a universe where almost nothing lasts, where stars burn out and galaxies drift apart and entropy eventually claims everything, the fact that one small creature found a way to loop back, to begin again, to refuse the finality of death, that's worth paying attention to. Not because it solves everything, but because it suggests that life is more resilient, more creative, more astonishing than we give it credit for. And if a jellyfish can break the rules, maybe, just maybe, so can we. We began by asking if one animal could refuse to die. Now, having followed Teratopsis through labs and theories, through molecular mechanisms and philosophical questions, we know the answer. Yes. but not in the way we imagined. The immortal jellyfish doesn't live forever as one continuous being, accumulating centuries of experience, growing wiser with age. It doesn't cheat death through perfect repair, maintaining the same body indefinitely, against entropy's pull. It survives by surrendering, by releasing its grip on what it was and accepting transformation into something new. The medusa dies, the polyp emerges, and in that cycle, that endless loop of death and rebirth, the organism persists. This is not the immortality we dream of. We want to keep being ourselves, just longer. We want more time as the people we are, with the memories we've accumulated, the relationships we've built, the wisdom we've earned through living. Turritopsis can't offer us that. Its gift is not continuation but renewal, not preservation but transformation. And maybe that's the more honest version of immortality, because nothing truly persists unchanged. Not stars, not mountains, not species, not individuals. Everything transforms. The question is only whether the transformation is gradual enough that we can claim continuity, or sudden enough that we recognize it as death and rebirth. The jellyfish chose the sudden path. We're stuck with the gradual one. But we're learning that the boundary between them is less fixed than we thought. Every time a research team successfully reprograms an adult cell back to a stem cell, they're demonstrating Turritopsis's principle at a microscopic scale. Every time a partial reprogramming experiment rejuvenates aged tissue without erasing cellular identity, they're walking the line between transformation and continuity. We're learning to speak the language the jellyfish already knows fluently. The grammar of dedifferentiation and redifferentiation. The syntax of controlled transformation. The vocabulary of genes that unlock specialized states and return cells to pluripotent potential. It's slow learning. We're prone to errors. We trigger cancer when we should trigger renewal. We erase identity when we meant to preserve it. We're students stumbling through lessons that evolution spent millions of years perfecting in Turritopsis. But we're learning nonetheless, and every experiment, every success and failure brings us incrementally closer to therapies that could meaningfully extend human health span, not immortality. That goal remains distant, possibly impossible, maybe even undesirable. But something valuable nonetheless. The ability to repair what's broken, to restore what's declined, to give people more years of health and function before the inevitable end. That's the practical legacy of studying Turritopsis. Not philosophical musings about eternal life, but concrete tools for fighting age-related disease, senolytics that clear damaged cells, partial reprogramming that rejuvenates tissues, enhanced DNA repair that reduces cancer risk, immune rejuvenation that keeps the body's defenses functional longer. These aren't science fiction. They're in development now, moving from animal models to early human trials, and while none of them will make us immortal, they might make the time we have significantly better. But there's a deeper legacy too, one that's harder to quantify, but perhaps more important. Turritopsis changed how we think about aging. Before we understood its transformation, aging seemed inevitable, a law of biology as certain as gravity. Cells divide, telomeres shorten, damage accumulates, systems fail, death follows. The sequence was fixed, the trajectory was clear. The jellyfish broke that narrative. It demonstrated that aging is a program, not a law. A set of instructions that can be paused, reversed, rewritten. The trajectory isn't fixed. It can loop back on itself if the conditions and mechanisms are right. This shift in perspective matters because it changes what seems possible. If aging is inevitable, there's no point in fighting it. You can only manage symptoms, delay the worst outcomes, make people comfortable as decline progresses. But if aging is a program, then programs can be debugged, edited, optimized. Suddenly, the question isn't whether we can intervene in aging, but how. Not if biological rejuvenation is possible, but what mechanisms enable it, and how we can safely apply them. Turritopsis gave us permission to ask those questions seriously, to treat aging as a solvable problem rather than an unchangeable fate. And in doing so, it joined a lineage of organisms that have taught us profound lessons about life by breaking rules we thought were unbreakable. Bacteria taught us that life could be simple, single-celled, yet extraordinarily successful. They've been here for billions of years, outnumber us by incomprehensible margins, and will likely outlast every complex organism on Earth. They taught us that simplicity is not weakness. Plants taught us that consciousness is optional, that you can sense your environment, respond to threats, communicate with neighbours, make decisions, all without a brain, without awareness, without subjective experience. They taught us that intelligence is broader than we imagined. Octopuses taught us that minds can be distributed, that intelligence doesn't require centralization, that neurons can be scattered throughout a body and still coordinate complex behavior. They taught us that there are multiple ways to build cognition. And now, Teratopsis teaches us that death is negotiable, that the arc of life from birth to death is not the only possible trajectory, that organisms can loop back, reset, begin again. Each of these lessons expanded our understanding of what life can be. Each of them revealed that the boundaries we drew, between simple and complex, between plant and animal, between mortal and immortal, were too rigid, too based on our own experience as one type of organism in one narrow slice of biological possibility. Life is wilder than we thought, more flexible, more creative in finding solutions to the fundamental challenge of persisting in a universe that tends toward entropy. Teratopsis is just one example. There are others. The tardigrade, surviving conditions that would kill almost anything else. Extreme heat, extreme cold, vacuum of space, radiation doses that would shred DNA. The hydra, regenerating entire bodies from tiny fragments, apparently not aging at all. The axolotl, regrowing lost limbs, organs, even parts of its brain. Each of these organisms has found a different solution to the problem of survival, and each of them offers lessons we can learn, mechanisms we can study, inspiration for technologies we haven't yet imagined. But Teratopsis stands out because its solution is so radical. It doesn't just resist damage or regenerate lost parts. It unwinds time at the cellular level. It goes backward through developmental stages that are supposed to be irreversible. It refuses the fundamental constraint that almost every complex animal accepts, that you grow up once, mature once, and never return to youth. The jellyfish says, I can return, I can start over. Death is a choice I make only when transformation is no longer possible. And in saying that, it asks us, what would you do with that choice? If you could reset, would you? If the cost was losing who you are now, would you pay it? For most of us, the answer is no. We wouldn't trade ourselves for extra time. We value continuity too much, identity too much, the person we've become through years of living. But maybe there are versions of the choice that are less absolute. Maybe we can borrow pieces of what Teratopsis does without committing to full transformation. Reset some tissues but not others. Rejuvenate the body but preserve the brain. Find a middle path between rigid identity and radical renewal. That's the work happening now in labs around the world. Researchers trying to parse the mechanisms into pieces, figure out which parts can be safely applied to humans, which parts are too dangerous, which parts might offer benefits without catastrophic risks. It's delicate work. The margin between rejuvenation and cancer is narrow. The boundary between controlled dedifferentiation and loss of identity is unclear. Every step forward requires caution, testing, verification that we're helping rather than harming. But the direction is clear. We're moving toward a future where aging is treated not as an inevitability, but as a condition that can be modified, slowed, partially reversed, where the decline that characterizes old age is no longer accepted as natural but challenged as preventable. Teratopsis won't be the only teacher in that future. We'll learn from naked mole rats, from rockfish, from bowhead whales, from every organism that's found a way to live longer or age more slowly than expected. We'll collect those lessons, synthesize them, create interventions that combine insights from across the tree of life. But the jellyfish will remain important because it represents the extreme case. The proof that aging can be not just slowed but reversed, that cellular time is flexible, that the trajectory can change direction entirely if the biology permits. Even if we never achieve full reversal in humans, knowing it's possible, somewhere in nature keeps the goal alive, keeps researchers asking, why not us? What would it take? How far can we push the boundaries? And sometimes that's what we need most. Not the answer, but the question. Not the solution, but the reminder that solutions exist, that biology is more capable than we assumed, that the limits we accept might be temporary rather than permanent. Turritopsis dohrnii is a very small animal. It fits on your fingernail. It lives in oceans most people will never visit. It does not think, does not dream, does not know that humans study it or what we hope to learn. It just exists, drifting through darkness, feeding on tiny prey, reproducing when conditions are right, transforming when death approaches, cycling through life stages that were supposed to be one-way streets. It's been doing this for millions of years, long before humans existed, and it will likely continue long after we're gone. Indifferent to our hopes and fears about mortality, indifferent to philosophy and ethics and the weight of infinite time, but in its indifference, it teaches. In its simple persistence, it reveals something profound about the nature of a life and time and the flexibility hidden in biology. It teaches that the rules can bend, that the impossible is sometimes just the untested, that life has more strategies for survival than we've catalogued, more ways of being than we've imagined. And it teaches us to look, to pay attention, to study the strange and obscure, because sometimes the most important lessons come from the smallest, most unlikely teachers. A jellyfish the size of a pinky nail has changed how we think about aging, mortality, and the future of human health. Not because it set out to teach us, but because we were willing to learn. And maybe that's the real lesson. Not about immortality or rejuvenation or cellular reprogramming, but about curiosity. About looking at the natural world and asking, what are you doing? How do you do it? What can you teach me? Because the answers are out there, in tide pools and deep ocean trenches, in extreme environments and ordinary habitats, in organisms we haven't studied yet and mechanisms we haven't discovered. Life has been experimenting with survival strategies for billions of years. Every species is a different experiment, testing a different approach, solving the problem of persistence in a different way. We've barely begun to catalogue those solutions, let alone understand them well enough to apply them, but we're learning, slowly, one organism at a time, one mechanism at a time, one small step toward a future where aging is optional and health spans match lifespans. Turritopsis dohrnii is one teacher in that education. Small, translucent, glowing faintly in the dark, doing the impossible with quiet consistency, refusing to die when death approaches, beginning again when endings loom. And in that refusal, that transformation, that endless cycle of renewal, it whispers a message we're only now beginning to hear. The boundaries are not as fixed as you think. The trajectory is not as certain as you assume. The future is not as predetermined as you believe. Life is flexible, biology is negotiable, and the rules that govern your existence, even the rule that says you must age and die, are just programs running on hardware that can, in principle, be reprogrammed. Whether we should reprogram them remains an open question. Whether we can do it safely is still uncertain, but Turritopsis has proven something crucial. It's possible. And sometimes knowing that something is possible is enough to change everything. We started by asking whether one animal could refuse to die. We end by understanding that death is more complicated than we thought, that mortality and immortality are not binary states but points on a spectrum, that the jellyfish hasn't defeated death, it's negotiated a different relationship with it, and maybe that's what we should be aiming for too, not defeating death, which may be impossible or unwise, but negotiating a different relationship with it. One where we have more time, more health, more years of vitality before the inevitable end. Turritopsis shows us the far edge of possibility, the extreme case, and in doing so it expands the space of what we can imagine for ourselves. Not immortality perhaps, but something better than what we have now, something closer to the lives we want to live, extended long enough to matter, healthy enough to be meaningful. That's the gift of the immortal jellyfish. Not a roadmap to eternal life, but a reminder that biology is full of surprises, that the limits we accept are often just the limits of our current understanding, that the impossible sometimes just means we haven't figured it out yet. The jellyfish has figured it out. Its solution won't be our solution. But in studying how it works, we're learning to ask better questions, and better questions lead to better answers. The conversation between human curiosity and natural ingenuity continues. Turritopsis has spoken. We're listening. And in that dialogue, the future is being written. One cell at a time, one experiment at a time, one small step toward understanding life deeply enough to extend it, improve it, and maybe, just maybe, transform our relationship with mortality itself. The jellyfish drifts in darkness, glowing faintly, infinite in its patient cycles, and we watch and wonder and slowly learn to speak the language of renewal it's been speaking all along. There's a small aquarium somewhere, maybe in Japan, maybe in Spain, maybe in a university lab you've never heard of. Inside it, in water carefully maintained at the right temperature and salinity, a jellyfish is floating. It's smaller than your thumbnail, translucent, almost invisible except for the faint glow of bioluminescence pulsing through its bell. 4 radial canals like thin orange threads, tentacles trailing into darkness, each one lined with stinging cells, ready to capture prey too small for you to see without a microscope. This jellyfish might be days old, or months, or years, or decades. There's no way to know. No rings to count, no markers that accumulate with age. It could have cycled through transformation a dozen times, or 100, each time dying as a medusa, reforming as a polyp, budding new individuals that carry forward the same genetic code in slightly different arrangements. The jellyfish doesn't know how long it's been alive. It has no memory, no sense of time passing, no awareness that it's doing something extraordinary. It just exists in the eternal present that all animals without complex brains inhabit. Responding to stimuli, feeding when food touches its tentacles, drifting with currents it can't understand or predict, and when stress comes, injury, starvation, the approach of death, it will sink, transform, and begin again. Not because it chooses to, not because it understands what immortality means, but because millions of years of evolution wrote that program into its genome, and the program runs whether the jellyfish wants it to or not. This is what immortality looks like, stripped of philosophy and meaning and human longing. It's not transcendent, it's not glorious, it's just a small animal, floating in darkness, following genetic instructions, persisting because persistence is what life does. But to us watching from outside, it's profound, because we know what we're looking at. We understand that this tiny creature has solved a problem we've been wrestling with since we became conscious enough to know we would die. It found a biological loophole, a way to reset the aging clock, a method for escaping the trajectory that claims almost every other animal on Earth. And in finding that way, it asks us questions we're still trying to answer. What would we do with that capability? Who would we become if we had infinite time? Would we be better versions of ourselves? Or would we stagnate, lose purpose, drift through centuries without direction? Would immortality make us wiser? Or would it just give us more time to repeat the same mistakes? Would it free us from the tyranny of time? Or would it trap us in an eternal present where nothing ever truly ends? Where consequences can always be postponed? Where meaning gets diluted by infinity? These are human questions. The jellyfish can't answer them. It just floats, glows, transforms, continues. Indifferent to meaning. Content to exist. Maybe that's the real lesson. That immortality without consciousness is simple. It's the consciousness that makes it complicated. The awareness of time passing. The knowledge that we will die. The fear of losing ourselves. The hope that we might persist. The weight of carrying our own existence forward through decades, knowing that every moment is bringing us closer to an end we can't prevent. Turritopsis doesn't carry that weight. It's light, unburdened, free in a way we can never be, because we know too much. We understand what's at stake. We feel the pressure of finite time, the urgency of goals we might not reach, the grief of relationships that will end. The jellyfish feels none of this, and maybe that's why it can be immortal so easily. Because for it, immortality is just metabolism, just the continuous operation of cells, responding to signals, following programs. There's no existential crisis, no philosophical burden, no question of whether life is worth living if it never ends. For us, those questions matter. They shape how we think about aging, death, and the possibility of extending life. They determine whether we pursue immortality at all, and if we do, what form we want it to take. And the answers aren't clear. They won't be clear for a long time. Maybe they'll never be clear, because these are questions without definitive answers. Questions each person has to answer for themselves based on their own values, fears, hopes, and understanding of what makes life meaningful. But Turritopsis gives us something valuable regardless of how we answer those questions. It gives us possibility. It proves that biology is more flexible than we assumed. that the constraints we thought were absolute are actually negotiable, that aging is a program, not a law, and programs can be modified. Whether we use that knowledge to pursue immortality, or just to extend health span, to live longer, or just to age better, is up to us. But the knowledge itself is transformative. It changes the questions we ask. It expands the space of what we can imagine. And sometimes that's enough. Sometimes just knowing that something is possible, that one animal, somewhere in the ocean, has figured out how to refuse death, is enough to change how we think about our own mortality. Because if a jellyfish can do it, maybe we're not as trapped as we thought. Maybe the trajectory from birth to death is not as fixed as it feels. Maybe there's room to negotiate, to adapt, to find solutions that evolution didn't hand us, but that we can engineer ourselves. We're not there yet. We're nowhere close to immortality, and it's not clear we should be aiming for it anyway. But we're learning. Every study of Teratopsis, every experiment in cellular reprogramming, every clinical trial of senolytic drugs or partial rejuvenation therapies, all of it is moving us incrementally toward a future where aging is less inevitable, less destructive, less of a constraint on how we live. That future might not include immortality, but it will include better lives, longer health spans, more years of vitality and function before decline sets in, fewer people suffering through decades of frailty and deterioration, more people getting to see their great-grandchildren grow up, to finish the projects that matter to them, to experience the things they've always wanted to experience. That's worth pursuing. That's worth the decades of research, the billions of dollars invested, the patient work of scientists, studying obscure organisms in laboratories around the world. And at the centre of that work, inspiring it, guiding it, asking the questions that drive it forward, is a jellyfish the size of a pinky nail, glowing faintly in the dark, cycling through transformations it doesn't understand, refusing to die not out of will or desire but out of simple biological programming. Teratopsis dorni, the immortal jellyfish, the animal that breaks the rules. It doesn't know what it's teaching us. It can't know. It's just trying to survive using the tools evolution gave it, following the instructions written in its genome millions of years ago. But in surviving, it teaches. In persisting, it inspires. In transforming, it reveals that the boundaries we accept are often just the boundaries of our current understanding. And maybe that's the most important lesson of all. Not that immortality is achievable or desirable, but that biology is full of surprises. That life has strategies we haven't discovered, mechanisms we haven't understood, possibilities we haven't explored. The natural world is a library of solutions to the problem of survival. Every species is a different book, written over millions of years of evolution, tested through countless generations, refined by natural selection, into something that works. We've barely begun to read that library. We've skimmed a few books, understood some chapters, missed most of the details, but we're learning to read, learning to translate the language of genes and proteins and cellular signaling into knowledge we can use. And every translation brings us closer to understanding life itself, what it is, how it works, what it's capable of, where the real limits are and where we've just been assuming limits that don't actually exist. Teratopsis is one book in that library, a slim volume really, a small animal with a simple body plan and a limited behavioural repertoire. But the chapter on cellular transformation is profound. The section on aging reversal is revolutionary. The pages on mortality are unlike anything else in the collection, and we're reading it carefully, slowly, with the attention it deserves. Because what we learn here might change not just our understanding of aging, but our relationship with time itself. We started this story with a question. Can one animal refuse to die? We end with an answer. Yes, but not in the way we imagined. Not through perfect maintenance or indestructible bodies, or conscious defiance of entropy. Through transformation, through letting go of one form and accepting another, through cycling between life stages in ways that were supposed to be impossible. The jellyfish refuses death by accepting change, by surrendering identity, by valuing persistence over continuity. We can't do that. We won't do that. We value ourselves too much, our memories too much, the people we've become through years of living too much to trade them for extra time. But maybe we can learn something from the principle, maybe we can hold ourselves a little more lightly, accept that who we are is always changing anyway, recognise that the rigid identity we cling to is partly illusion, partly fear, partly reluctance to acknowledge that we're already the ship of Theseus, already being rebuilt piece by piece, already different from who we were yesterday. And maybe, armed with that recognition, we can approach aging differently, not as an enemy to defeat, but as a process to understand and negotiate with. Not as a fixed trajectory, but as a program we can learn to modify, within limits, within ethics, within wisdom. That's the conversation Teratopsis invites us to have. Not about immortality specifically, but about what it means to be alive, to persist, to change while somehow remaining ourselves. It's a conversation we're just beginning, one that will take decades, maybe centuries to fully explore, one that will require not just scientific understanding, but philosophical clarity, ethical frameworks, social consensus on what we want the future to be. But it's a conversation worth having, because at its heart is a question that matters to everyone. How do we live well in the time we have? Whether that time is 80 years or 800, the question remains the same. And the jellyfish, floating in its aquarium, glowing in the dark, cycling through transformations we're only beginning to understand, reminds us that the answer might be simpler than we think. Let go of what no longer serves you. Transform when transformation is needed. Persist through change. Begin again when necessary. The jellyfish does this at a cellular level. Maybe we can do it at a psychological one. Maybe the real immortality isn't biological at all, but the ability to continually renew ourselves, our perspectives, our purposes, our connections, so that we remain vital regardless of how long we live. That's not what Teratopsis teaches directly, but it's what we can learn from watching it, from studying how it survives, from understanding that persistence doesn't require rigidity, that continuation doesn't demand unchanging identity, that life is more about flow than fixity. The jellyfish drifts, glows, transforms, continues, and in that simple existence, it asks us to reconsider everything we thought we knew about aging, death, and the possibility of renewal. We don't have all the answers yet, we might never have all the answers, but we're asking better questions. And in a universe where almost nothing lasts, where entropy claims everything eventually, the fact that we're asking, the fact that we're learning, exploring, trying to understand, might be the closest thing to immortality we'll ever need. Because ideas persist even when individuals don't. Knowledge accumulates even as generations pass. The conversation continues even when individual voices fall silent. Teratopsis has added its voice to that conversation. Quiet, wordless, expressed only through the language of biology, but clear. Nonetheless, the rules can bend, the impossible can happen, and life, in all its strange creativity, is far more flexible than we ever imagined. That's the message. That's the gift. That's what the immortal jellyfish teaches to anyone willing to look, to listen, to wonder at the small glowing creature that refuses to accept what almost every other animal on Earth accepts. Death is not inevitable, at least not in the way we thought, and maybe that changes everything.